Sitar Bates for the kind invite for me to share some of the views. It's a pleasure to be here, and I just over time I understand that the objective of this particular event is to enthuse young stars, young students who are on their way to build their careers, and share the some insights of the world of scientific innovations and the vehicles to build those innovations, the path, the journey to build those innovations. So I will try to share this today very quickly. Details are just for examples, are not necessarily for anything else. So friends and young students, it is more of you should get the feel of what's going around. And I'm happy to take questions. So I will try to do, what I'll try to do is that go through some of the details from my side very, very quickly. And then allow you to possibly, interact and ask questions based on. So let me start by sharing my slides. Please let me know if you can see my slides. Can you see my slides?
Yes, Professor.
Yeah. Okay. So, as you know, the larger aspect of the perspective is to what are the current ecosystem in terms of scientific entrepreneurship and innovation and building a sustainable future. And I have built it from a perspective of CCAM, the work that we are doing here. So just to give you as something that I use significantly to begin my talk, we all know about scientific entrepreneurship. But before I go on to talk about scientific entrepreneurship, let's talk about innovations and how innovations are built for solving societal problems. So scientific knowledge reaches its maximum impact when it solves societal problems. We all know about it. We uncover, we discover, we understand more of science around us, and we use that knowledge to solve societal problems. We have several problems, several examples around us of such scientific journeys across countries. You would think of many scientific entrepreneurship journeys that you know of. Globally, the people who have built life sciences, let's talk about it, from drugs to therapies to diagnostics in India. Many such are there, like Ciplas of the world, Biocons of the world, Zydus Cadila, many others. These are all science-based entrepreneurship, building enterprise to take scientific knowledge to the next level. This knowledge could be inbuilt or built collaboratively. That's perfectly all right. So when a scientist decides to take her or his work further towards building a technology or product that they believe in, this is when the scientific entrepreneurship journey starts. In India, in last 8 to 10 years, it has been a phenomenally productive environment to promote science-based entrepreneurship. A lot of work has been done. And entrepreneurship is a journey. It's a vehicle to build innovations, finally, that can solve the problems around. It's very important that you build something that is beneficial to others. That's where the impact comes. You can always build something otherwise. You can always build 5-legged chair. You can always build 10-legged chairs. You can always build anything that you think is exciting, but if it is not beneficial to anybody, those fantastic innovations will die its own natural death. They will not make an impact. And that's why connect of innovation with solving problems, benefiting society at large, is a very important connect, and you have to understand it. And entrepreneurship is a journey for that. Let me go further. So just to How CCAM is involved in supporting that environment as an ecosystem player. So CCAM is a part of the Bangalore Life Science Cluster, which has other two organizations like NCBS, National Center for Biological Sciences, a top-notch, premier-most research organization in biological sciences, a center of TFR, which was the original institute which started the campus, and then INSTEM, which is Institute for Stem Cell Biology and Regenerative Medicine, and CCAM, that is Center for Cellular Molecular Platforms, came together, and together we formed what we called as a Bangalore Life Science Cluster. The Bangalore Life Science Cluster at CCAM, we have two broad verticals. One is a technology platform, and another is entrepreneurship programs. And very quickly, to just give you a perspective, when you do science, you have a lot of technologies that you would require to take your scientific idea, test it, and validate it further. And this is also useful in research aspect. So we have state-of-the-art, global state-of-the-art technology platforms which allow researchers from startups, industry, and academia to come and utilize these resources and expertise to take their scientific concepts further. So we have flow cytometry, high throughput screening, genomics, microscopies, mass spectrometry, structural biology technologies, micro devices, and transgenic animals. There's several of those around. And through this, we have made a broad impact across India. So this is enabling science, enabling scientific ideas to go forward. So this is a technology platform vertical. But at the same time, something that I will talk more about is an entrepreneurship vertical, which has the ability to fund, incubate, mentor, and accelerate new ideas. But more importantly, it's very important before we do all that. These are just the, our abilities. More important is to spot good science, which is before what you would say the funding. How do you attract good ideas to come to you? How do you engage with those good ideas? How do you then assess, massage, improve those ideas with the original inventors of those ideas, and then help them through these machineries? These machineries are not assets of an organization. These machineries are the means of taking an idea towards the building to an innovation all the way. So these are the means, but very important is a mandate of spotting a good scientific idea, helping that idea to go through through these machineries. It's a very important thing. So what's happening across the thing? We have some very, very good innovations that we are actually have supported over last 8-9 years now. This is just a view, and I will not spend too much of time on this. We have around 250 to 300 funded startups from CCAM through our collaborations with different organizations like DBT by RAC, like Niti Aayog AIM, DST, MIT, Government of Karnataka, many people who have supported our efforts. And just to give you a perspective, we have now cohorts of innovations sitting out in different domains like antimicrobial resistance, regenerative medicine, medical devices, non-communicable diseases, very important, maternal child health, infectious. We have now a large number of innovations, which as an ecosystem, we are supporting them through, as you saw, funding, incubation, acceleration, and so on. And of course, there are life science has impact in agriculture. We have an environment so that you would have a waste management and bioenergy, veterinary sciences, nutraceuticals, industrial biotechnology, and so on. Some of the areas, while we do that, and some of them are really, really groundbreaking work that has happened in this, but some of these areas are important for India, like maternal child health. And we know that broadly, India unfortunately doesn't rank very highly in terms of the maternal child health index. We have many things to improve. Many people and many young lives and many maternal lives are even lost, and we have a lot to improve. So we are working to see how uh, those innovations can actually impact those things. And I'll give you a quick example across a few things. But so let's begin with a very important thing of fetal monitoring. So if you know, fetus monitoring is to understand when there is a pregnant mother who is expecting, how to understand the fetus monitoring, fetal heartbeat, fetal movement while it is there. Uh, Usually, these are not available until you go for something like an ultrasound and so on, and which happens once in a quarter and so on. But this is also only possible in urban settings, in cities. But possibly it's not easy to do it in non-urban setting. And these are all things that you have to spot early. You have to detect early in any irregularity in terms of fetus growth. And this is where simple devices, which are wearable, a small wearable device, which can be available with primary healthcare center, which can be available with a district hospital, or which is possibly available, for a rent, which can be just worn by the expecting mother, and the data can be seen by, treating doctor to ensure that the all the details are fine. Look at the impact of such a small, small intervention, but as a large impact, so that you don't have to buy ultrasound machines only for that, or you don't have to access, because we don't have ultrasound machines across all the hospitals. So, and think about how many people, which would be crores and crores of people who do not have access to that. So such innovation is very, very important. I'll give you another example. I'm just wary of the time, so I'll not spend too much of time on everything, but give you another one is this diagnostic platform for neonatal sepsis. Now, excuse me. Now, this is another issue. So, as you would all know, neonates are the very young babies, like immediately after birth, in a way, very early days of their life. Now, many times, these neonates or the young babies after birth, they have infection, so they have fever. And as you know, when you have fever, you have to understand what has caused the fever. And for that, you will have to kind of understand how, so you sometimes you have to do their from the foot, you have to do the heel prick, take some, blood, and do the understanding of either infection or also in terms of how are they responding in terms of immune response from a inflammation, like cytokines and so on. From young babies, as you can imagine, you cannot take too much blood. So there is a restriction because they're very, very young and you cannot just pick them again and again. So this particular work for the diagnostic platform, we are about with our one of our company, we're about to launch it this month, if not this month, next month, a device which doesn't require blood from neonate, but just a saliva test where you can actually take saliva and get the same amount of understanding of how the baby is responding to the treatment that is given by understanding cytokines, possibly, CRP and others which are there. I'll not go into technicalities, but the doctor can take just a saliva swab, which is easily available, non-invasive in nature. You take the saliva and then you test it and you understand that baby's inflammation response is going high or it has come down, and if any further intervention is required. This will be possibly first in class in the world to actually establish it. So we are very, very proud of this. There's another one, and I can keep going, friends, but I have to be careful of time. But the third one is for device for neonatal respiratory support. I'll give you a very quick example on that as well. So device for neonatal respiratory support. So many times when the babies are born, and if they are premature birth, their organs are also not fully developed. And in that case, when organs are not fully developed, even the lungs capacity is not there. The breathing respiratory system is not fully developed. And hence they need support early, at least few early hours or a day or two, to give the respiratory support, breathing support, which can be very simple. But these devices are there. These are there for many years, but these devices are built by international companies and they are fairly sophisticated. They're very expensive and they are possibly so sophisticated that they not work in a semi-urban or a rural setting. So this particular company, in Excel, has developed and now in market where they have devices which can provide this respiratory support, which is called a CPAP device. I'll not go into details again, but this device is now approved by the regulatory authority and for last year or 2 years, and including in COVID, has been able to give for respiratory support for neonates and even children per se. And we have gotten notes from neonatologists that they have been able to save lives using such devices. And this is what the innovations impact is all about. I can keep going on that. This, this is another device that we are about to launch in a month or two, which is for neonatal hypoxic ischemic encephalopathy, which is nothing but birth asphyxia. Again, a device, neonates, which more or less sometimes prevents the lack of oxygen, which happens early after birth, which causes the neuronal damage or a developmental damage because of the neuronal damage. And how do you control that component? How do you provide the temperature control? How do you provide the enough environment for a proper oxygen support and so on, which could prevent the neuronal damage after the young baby? These are all developments which have happened over 5 to 6 years. These are not recent, but this is this ecosystem which have identified these ideas, supported those ideas, not by funding. Funding is good, but is only one piece of the larger puzzle. It's all about hand-holding those ideas. It's about understanding, validating those ideas day after day by different stakeholders, by regulators, by users, by beneficiaries, by doctors, by physicians, by many people, and ensure that this is something that society needs at large. And that is something very, very important for now. I just give you a few examples, friends, in maternal child health only. If you remember, we had many such cohorts, and I can keep on going, but this is where the importance of building innovations come together. What we are doing together is actually building innovations in these areas. So we have maternal child health. We are doing similarly in agriculture, where we are attempting to build a lot of innovations for the higher crop production. The governments have very ambitious plans of increasing farmers' income, and crop production is a very important aspect of, from the same amount of land, same cost, can you have a higher crop production? And there are newer scientific interventions which can be done for a higher crop productions to contribute to the higher income to farmers. So that is one. The second one is something where farmers and farm laborers are constantly exposed to pesticides day in, day out. And that causes chronic pesticide toxicity, because and over years, it becomes very, very, damaging to the health and many times deaths as well. So how do you have a new nanotechnology-based solution which can actually prevent the pesticide absorption at the skin or the breathing level? So how do you have devices which can prevent the toxicity, even though if it is used? So that is a new, that product is also there, which is there. Of course, the other ones are, novel bio-based antimicrobials for plant pathogens. We are using pesticides, but there are other biopesticides which can control, because pathogens have to be controlled. A lot of chemicals are used, which are not good for many reasons, of course. So do you have a natural pesticides, biopesticides, which are not harmful in a broader setup? So that's also a very, very important component per se. And there are much more capabilities that come in. On the larger aspect is, uh, as one would call it, and antimicrobial resistance. Uh, One of the aspect, as we all know, is, and we are hearing it, and I hope you must have read it. Coming from a biological science background, AMR is a global challenge, and how it's a it's a it's a tsunami as a that is coming as a silent pandemic that is going to cover the entire world, which has possibly covered the entire world, and going to increase further. So we need to build innovations for prevention of AMR. So we have several innovations in the field of AMR. At the same time, early diagnosis, if that is to happen, and also build new therapeutics so that even if there are resistant bacteria which are there and which people die of because there is no antibiotic which can cure that infection now, how do you build new antibiotics or antibacterials to possibly address that? And we have several solutions which are doing very, very well. We have one solution which is coming from a company called Bugworks. They have just raised around 135 crores or $18 million, around possibly around those numbers, to go into phase 1 and phase 2 for the clinical trials of the novel antibiotic for gram-negative bacteria. We have another company called Kangajan, which is using bacteriophages for the same aspect in terms of control of the resistant bacterial infections. They hopefully will also be possibly going for the phase 1 in this year or later this year or possibly early next year. So again, there's a scientific big innovations which are supported by the ecosystem, which actually makes a larger impact and some of the work that is there. At the same time, I don't want to really go into further details, but when you build this ecosystem, it also creates platforms for us to respond to challenges like COVID-19. Think of it, if we did not, friends, if we didn't have such platforms, there wouldn't, how would we come together and respond to something like COVID-19? So in such scenarios, I would say that we have been able to come together as a platform with academia, industry, and all to, excuse me, to more or less what we call it as respond to the need. And one of the earliest needs in the COVID-19 was diagnostics. We didn't have enough diagnostic kits when we started, and testing was important, as we all know. So what we wanted to do is that at that time, that can we build now kits from India, which can be at the same quality as a global standards, but also cost effective. And just to give you an outcome of this, we work with around 150 to 175 companies through these platforms, which are supported by Rockefeller Foundation and DBT. We have been able to now have capacity to produce 10 lakhs RT-PCR kits per day through our companies, which we have supported through this platform. Not only that, if you remember the earliest COVID-19 RT-PCR kit, the original cost was around 4,500. Currently, we have been able to, all of us have been able to go out and get the RT-PCR in 300 rupees per se. Original cost may be only 50 rupees now. This has happened because we have been able to build high quality at a higher scale. So larger supply has brought down the cost as you scale up. So this is a great example of an ecosystem which can bring people together and respond to such challenges. Similarly, we had a platform for what we call it as Innovation Deployment Accelerator, to bring together innovations which were there, but not really, but had some distance before they could be used in COVID-19 setting. So we we assessed around 1,100 innovations over time in the first in the April month of the 2020, and decided that these are the innovations which have possibility and potential to now support India's response and the global response to COVID-19. And very happy to share with you that 23 out of 30 innovations that we have selected are right now serving people in hospital setting, primary healthcare setting, or other settings. And we have deployed them ourselves in 17 states of India. So this is an impact of an ecosystem, how we want to grow together and build it at large. So just to conclude, I'm sorry about my recurring cough, but so scientific entrepreneurship is crucial for countries like India. And you just possibly saw the reasons to build its own innovations for its own problems, because every geography has its specific problems. There are many models to build science-based innovations, be it through conventional entrepreneurship model that we talked about, or a mixed translation model. But what I wanted to leave today is with that exciting time. This is an exciting time for young scientists. If you have your own interest in building innovations and with aim of solving societal problems, this is a very exciting time. If you have some ideas, test those ideas and build it through the ecosystem which is available. I only give the perspective of CCAM, but there are fantastic examples, including the efforts of Sitare and the National Innovation Foundation and the Gyan and other fellowships which are given. I think it's Gati and all that. So I think this is fantastic opportunities for people to look at those ideas that they believe are exciting and how they want to now build it further. Talk to people, hear them out about your innovations or your ideas, and then build it further. What I will do is that possibly I'll stop today for this, but I do not want to go before saying thank you all for patient listening. If there's any need for any further information, details required, please feel free to reach out at this coordinates and we'll be happy to respond back. Thank you so much. I'll give back to the session chair. Thank you so much.
Thank you, Doctor. Thank you for your wonderful speech today. Your speech may be inspiring for the students to take up the entrepreneurship and to develop their own innovative ideas. Students, if you have any queries or you want to ask something to Doctor, you can ask.
We need one student to begin so that others can, kind of come out as well. So somebody has to take a lead and so others can come out.
No one is there? No questions? Just feel free to ask.
Good morning, sir.
Morning.
So my question is that which field, according to you, is the most growing field right now in biotechnology?
So I would say that it's a I think it's a very good question, but let me turn around a part of it. Which are I would say that most futuristic and advancements in biotech, rather than saying the field per se? Where are some of the most exciting innovations happening? We already talked about, or we are rather talking about globally, is how people are going to do with a nucleotide-based mRNA or a DNA vaccines. That is a very important thing. That is very, very highly emerging and futuristically. Could that be your, way into the therapeutics? That will be a very exciting thing. Cell therapy and stem cell therapy is another very, very exciting thing. It has a potential to solve many, many healthcare diseases. We have heard for many years about gene therapy, but cell therapy seems to be much, much more feasible as we go forward. I don't know whether yesterday any of you read, globally third patient got cured of AIDS by stem cell therapy implant. The patient was, if I'm not wrong, a leukemia patient, but because they got the stem cells implant from an individual who was resistant to HIV, now the leukemia patient has been cured of HIV. But this has happened because of the stem cell implant that happened. But futuristically, implants could be for which I didn't talk about today. We have many companies in stem cells for vision disorders. Futuristically, many other disorders will be addressed by stem cell and cell therapy. So that will be another exciting thing. Third is going to be definitely gene editing, something like CRISPR and the future versions of CRISPR that we all know about, not only in healthcare, but many other fields, including agriculture, diagnostics, therapeutics, and research aspect. I would say that other than that, I think in terms of the delivery, anything that allows remote health assessments, that will be another major change in terms of healthcare. So I would stop here. I just summarize it for all. I mentioned nucleotide DNA, mRNA based therapeutics or prophylactics. B, stem cell therapy. C, gene editing like CRISPR and future versions of CRISPR. And 4, I mentioned about the remote health assessments. Those will be very important. So I would just say that there are many, but these are the ones which are going full force in terms of addressing global challenges in healthcare, agriculture, and otherwise. Thank you for your question.
Doctor, there is some question in the chat box.
Yeah.
Hope you can answer.
So some Rakshit Gowda from Maharaja College, Mahajana College, Mysore, saying vaccine production for virus is not easy as for bacterial infection. So what are the challenges we want to face? We want to face for vaccine production for virus infection. So I don't know, just if I understood the question right, what Rakshit is asking is that the challenges of vaccine production for viral, but this is what exactly has happened around us. The vaccines which are developed by at Oxford from AstraZeneca, which is in the shape of Covishield for us in India, or the one that has been developed by Bharat Biotech in Covaxin, or many other vaccines, Moderna's and all those. These are all vaccines for the viral infection here for now, COVID-19. Idea is that you need to have a vehicle to generate a kind of a immune reaction so that when in future, the viral infection happens, it is already recognized, your immune cells already has a memory of something that they have been exposed previously and take care of it faster than otherwise. So I think in bacterial infections, people are attempting such vaccines as well, but it is both at the basic concept level, it is the same. How do you give a small primer or just a quick non-infectious version of or a part of that so that your body recognizes it, starts the immune reaction, but it is not infectious, it will not cause you harm, but it will keep as a memory. And when the real infection, if it ever happens, your body has a memory of it, hence it can respond it back to it. And that's what the idea is now. Based on the approach, whether it's an mRNA vaccine, so the challenges in terms of production of mRNA, specificity, and so on, or be it an inactivated viruses. There's a different ball game, how you produce. One you have to produce in BSL-3. The other one is just a nucleotide sequence. You do not possibly need a BSL-3 to handle that. So there are different infrastructure and production challenges. And I hope that is the question that you were asking. But I just and the answer is satisfies your query. Thank you, Rakshit. There is another question by Tarumoy. Thanks, Rakshit, for confirming. Tarumoy Das from NIT Durgapur. Is when new drugs are developed, if in the clinical trials it fails, what are the next steps towards it? Okay, so it's a very good question. Give me a minute. I'll just charge my computer 1 second. So I think Tarumoy is asking whether if in case the drugs fail in the clinical trials, what happens? So there are 3 phases of clinical trial. Clinical trial 1, which is more of safety, whether whether the administration of this drug causes any major side effects to humans. That is safety trial, clinical trial 1. Then you have an efficacy trial in 2, and a larger trial, which is 3. So what you're saying is that the safety is there, whether it is effective, and a larger unit also it is effective. Now it fails for whatever reason. Of course, it cannot go forward because it has failed the criteria to be taken out by the regulatory body. But in terms of development, it has come this far. So the company has an opportunity or an option available to work and understand what caused the failure, either in efficacy, that is effectiveness, or the side effects. What could have caused, so they can redefine it and develop it further. Now, just another interesting angle is that many times, and as we now understand, not all drugs are respond equally, or are actually, or not all human beings respond to the same drug equally, because we are different human beings, we have different microbiome, we have different genome, and all that. So now what people are doing is that to understand that, okay, this failed in a larger cohort, but is there a specific kind of small group, Hispanic group, you have this group, South Asian group, whatever, genetic or microbiome based and all that, where this works better. So now this is called precision medicine, where the drug has a larger impact for this kind of people, either because of the genome, microbiome, whatever others, or kind of same cancer, but which behaves differently in this group. And it could be ethnic group, it could be other kind of group, it could be age group, it could be gender based, and suddenly the same drug in a smaller group or a specific group works better. So you have to identify that, understand that, oh, in a larger group it didn't work, but in a small, if you just take small groups of the larger group and say, oh, it didn't work in 100 equally, but in this 20 it worked phenomenally well. So can we now treat it as for this one and start the process again? So there are many versions. You have to read all those details carefully to understand, and that's the option. If you think if it has failed miserably, then companies have decided to close it completely. So I hope I answered that question, Tarumai. Okay, thank you.
One more question has come.
Okay, surely. Can this be a last question and then I jump out if it's okay?
Yes, doctor.
Yeah. So Pratyusha Gaura from VFSTR asks, can mutations in virus or any particular microbe can make the pathogenicity of it less severe? Because as we observe, the symptoms of COVID got somewhat less in third wave when compared to first wave. Yes, it can go both ways, and we know that, right? So any mutation can have different effects, but let's possibly not connect to the COVID, but you can imagine a pathogen is there. If it mutates, right, it changes, something changes. Sometimes it becomes much more severe, sometimes it becomes less severe for whatever reason it may be. Many times it mutates, as we know, is based on largely for a better survival. Sometimes people say this virus wants to become less severe so that it stays in, it can grow. Influenza is less severe, hence it is growing. Every one of us get influenza, right? it is less severe. But think of it, it is a dominating virus. It comes to us many times a year, all of us, right? And that's what it is. So sometimes this evolution game happens when for a long longevity and for other things, it becomes less severe so that it stays around us, it grows happily. And it's all about survival. It's not about killing your host, or it's keep putting your host in a bad situation. It's about survival for itself. So it's a pathogen which requires a host. It is better for a pathogen to keep the host alive, to keep the things good, and then spread as much as possible. So mutation can cause both ways. However, the point that is in the third one, it also, if you see carefully the data, it is also because the same third wave, that is Omicron, has caused different level of severity in different geographies. And some of the data that comes from US suggests that there is a huge difference between people who have not been vaccinated and were not previously exposed. Severity is not very less. It is there, not very high, but it is not also very less. So it's a response to how your people who have been vaccinated, how it became much less severe for many people. And so there are many factors. It is not only one, but there are many factors. I hope you answer your question. I hope I answer your question, Pratusha. Thank you.
Thank you, doctor. Thank you for your valuable time.
Absolutely. Thank you. It's very good to be here, and my best wishes for the entire group, as well as to the entire organizing committee, including Professor Gupta. My best wishes for all. Thank you so much. All the very best.
Thank you. Thank you, doctor.
Bye-bye.
Students, please stay online. In another 5 minutes, we will start the next session. Okay. Welcome back, students. We'll start the next session. So in this session, we will just give an overview about the SRISTI lab facilities and the products developed by SRISTI for various applications like agricultural, veterinary, pharmacology, and microbial applications. Okay. So we have with us Dr. Premlata Pati. She is a senior scientist and a GYTI coordinator also. She will present us about the SRISTI's lab facilities, and will, because most of you will be coming here to work, right? So for your projects, so you can get us an overview. If you have any queries or questions, you can ask Dr. Premlata. Dr. Premlata, you can start.
Yes, thank you. Good morning, everyone. So I just want to see the video of the students. So can you switch on for a while? Yes. Okay. So myself, Dr. Premlata Pati, I'm a senior scientist here, and also I'm coordinating GYTI of BIIS. So I will tell you about our SRISTI lab today. Okay. So before that, I just want to share something that we have. I'm not seeing the whole, 65 participants we selected, but your attendance is only showing that you are 34, 35. I don't know whether all are attended the session or not. So that we will discuss later. So I just want everyone should listen all the lectures, because this Saturday we are going to make a quiz for you. Okay. So before that, we are not sharing any any PPTs to you, so that how can we know that you are attending the session? Because the PPTs will be with you and the people will write that things. We just want to be attentive. Each session will help you later, right? So let me start my topic today. Okay, so I'm just telling you, this is SRISTI's lab facilities and activities. As you know, when you applied this program, so you read the guidelines that you will attend this workshop, and you will give the assignments, and we will share, and the course schedule is also, I think, with you, that you are going to learn about microbiology, pharmacology, agriculture, veterinary, and all aspects we are working on in biotechnology field. We are going to learn all these things. So what is our lab's motto? So when you 65 students, you will be assessed by your assignment, your quiz, and your performance in this workshop. Then we are going to select 10 students for award. Those students will be awarded. They will choose that whether they will come to our lab, or due to COVID situation, we are also allowing them that they can do their work in their home institute as well. Okay. So when you are coming here, so you should know, sir, what facilities we are having, we can give you. So you should know. So that things I will share to you. Okay. So before that, I want to tell you, so how there is a very good story about our lab, that how this lab established. So you know that in the first day session, Professor Anil Gupta sir, he introduced about the honeybee network, and the SRISTI and the GYAN, and also and about the SRISTI laboratory. So there is a very good story in the establishment of this lab. So I just want to tell you shortly, because our lab was established 22 years back. That means in 2000, in the year 2000. So it was a story when Professor started. In this slide, you can see the honeybee network. He started in 1989. Okay. Then in this network, he just started that Srishti in 1993. And another branch also network he started in 1997, that is Grassroots Innovation Augmentation Network. So when these three things were already established, so professor was always thinking that it was his dream that how this, because he was doing the Shodhyatras, he was doing the traditional practices, he was collecting information from the rural areas, from the farmers. Okay, so that there is many informations. I'm not audible. Okay, am I audible properly?
Yes, ma'am.
Yes, doctor.
Okay, okay, fine.
Just go to full screen, I think.
Full screen is open, I think.
Yeah, okay.
Yes. So our lab's full name is Sadbhav Srishti Sanshodhan Natural Product Laboratory. So what is this Sadbhav? Because you people already know that this is Srishti Laboratory. So you should know the story, how it became. So it was 22 years back that when Professor was in Srishti, one stranger came and he said, we just browse about Srishti and we got information that you are doing these things. So we are a trust that is there from Mumbai, the Sadbhav Trust. Okay. So they just asked simply, Professor, that is there a dream that has not remained unfulfilled? So professor was just surprised how a stranger came and he is asking, because it is already in my mind that there should be a laboratory like that. But how this coincidence happens? Anyway, that happened. And professor just explained, yes, there is dream that is always in my mind that Srishti, we have lot of innovation of traditional knowledges and through volunteers of Honeybee Network. But while trying to work with scientists, we have not been able to try the grassroots knowledge and experiment practices without adding the value. Because you know, if you are going to any village or any rural area, but still the farmers are working now, they are working. They know how to treat their plants. Even in the rural area, they don't have that much hospital facilities, but still they know how to heal the disease. But professor thought that this is science, but nobody is recognizing that, right? So he thought, why will not make a laboratory which will be something different from any other laboratories? Just like when you were hearing that about what is a laboratory, what is a research laboratory? We only know that there will be some equipments and some research, some, you know, what we read in our colleges, universities. We will apply these things in the laboratory practically. But this Srishti laboratory has some different purpose. Just like, you know, in your laboratory, you are working something, then you are publishing paper and you are trying to take it to a clinical level or in a market. But here, what is our purpose? That if a farmer is telling something that my field is affected by some pathogens or something, some insects, but I tried to recover it from my own knowledge. So professor said, so why you are not sharing your knowledge to us? Then he started documenting the things, and we are publishing that things in Honeybee books. Then he shared that things in the laboratory, and he collected the scientific persons that this is a farmer said that he tried this practice and he got good results. So you scientific team, please give value to that practices. So he is the innovator, but nobody recognize him. So you scientific people will work that what actually behind that practice, which is effective. But this can't be go to market directly. We are the person here. We will try to make that innovation to market or commercialization, right? So this is a reverse process. We are not going from lab to market or field. We are coming from field to lab, right? So this process we are continuing. And in that discussion, suddenly a quick proposal developed and they established this Sadbhav Srishti Sanshadhan Natural Product Laboratory. So this is the overall story of our lab. Yes. So this is our Srishti Bionist or Srishti Laboratory. Those students who will be selected, so we wish they will come here and in our lab they will work. So today I will just give you the lab overview that what exactly we are working, because what facilities we are having, because sometimes you are undergraduate students now. So not all students, all colleges having all facilities. Okay, but still the students wants to do some good research. For them, our lab is open and you come here and see the facilities and explore the facilities. So what works we are doing? So first, before that, I will say the objective. What is the objective of our work? So first is we are doing the Shodhyatras. Shodhyatra, it is a Hindi word. So you know that what is Shodh? Shodh is research. So from our lab, each year, a group of people and group of team, and there are many more volunteers are involved here. So they are starting from here and they are just roaming around all over the country. And they are going to different places, just like they mostly prefer to go in the extreme environment conditions. Okay, means if it is summer, you know how hot is summer in desert areas, but they prefer, these people prefer that we will go to that area and we will collect samples, that because these samples are very, they are growing in a harsh condition. So we definitely get some good compounds or good results from that area. So in the winter, heavy winter, they are going to Jammu, Kashmir, Ladakh area, that they will collect sample from that area. So in Shodhyatra, we are collecting samples, soil samples and some other samples. And we are also interacting with local farmers and the local peoples, that how they are managing all traditional practices. And we are documenting that things, then we are coming to your lab to validate that things. Okay, then is validation and value addition of the grassroots innovations that I shared now. Then we are preparing here development of eco-friendly.
Excuse me, ma'am. Sorry to interrupt you.
Okay.
But ma'am, the presentation of your slide is freeze. It is still on the first current slide.
Is it?
Doctor,
Yes.
Okay.
check your slides. It's still showing the first slide. It's not moving.
I don't know why it happens. So is it working?
No, not.
So let me share again. Huh?
Ah.
Is there any stop share? This is right. Is it?
Yeah, now it is okay.
Okay, okay. Sorry, because the network is very poor here. I don't know because of that. Okay, now you can see.
Yes, doctor.
Okay. So next is development of eco-friendly non-chemical options for pest management and animal healing. As we told that we are going to tell you about the agriculture practices and veterinary practices. We know there is medicines and pesticides for everything. Now solution is there. But our purpose is that how it could be, natural. We will try our best to avoid the chemicals, because by adding the chemicals, definitely we are solving the problems, but there is some side effects. So our purpose is that the traditional things, because you just imagine a farmer in a village, so he is also a very poor person, so he is not he can't afford some costly chemicals, but still he always finds a solution that how I will treat my field from the insects and pests. So that things, uh, we can make a product. Okay, so that is our purpose, that okay, that is very organic, and we can use these things when it is showing effect in plants or any human disease or whatever. So why we will not try that practices? Definitely it will be less chemical as compared to other market drugs or market chemicals, market pesticides. Okay, the product things will be um, given to you. The description will be shared by our another scientist. So that will be the latter, my next presentation you will see. So in short, this is our objective, that we are working on the product development of non-commercial, non-chemical pesticides. And as Professor said in the first day, you remember, so our purpose is to bridge the gap of the formal and informal knowledges. Okay. So my slide is now visible.
Yes, doctor.
Okay. So these are the research areas we are working on. So currently we are our ongoing lab activities. So this is the phytochemical screening. Phytochemical screening means we in Srishti have our medicinal plant database. So we are extracting the compounds from that medicinal plants, and we are checking its screening, that what phytochemicals are available. And we are also doing the activities of antimicrobial screening. Then development of botanical and microbial pesticides. So we are also working on the the plant pesticides, also the microbial pesticides, and we also work on the veterinary and human formulations. We have previously we have developed a lot of products, and for that developing that products, we are working hard here, that how it will be come in a product form. Then also we are screening antimicrobial leads, also from medicinal plants, and also from the microbes. And also we are working on affordable diagnostic devices. In Srishti, we developed some microfluidic devices, paper-based devices, for blood grouping, and also for, in cattle, the mastitis is a major problem, and detecting that mastitis level, we made a devices, and we are already worked on that, and also currently we are preparing that devices to sell in the market. And we also have a good molecular biology and cell line development facilities. So when any bacteria or fungi is showing any good results, so we are just isolating the DNA, and we are also we are also developing some cell lines. And in our future prospects, because now you know that when any drug or anything is coming to market, it takes a long period. But nowadays the science has developed a lot. So everybody, I think you know about the in silico drug designing. And I would say that because of this in silico drug designing, within a year we got our vaccines. Because if you start from testing that any drug isolation process in wet lab, it's sometimes taking time. However, it gives the hint, the in silico process, so that we are also working on that, that if we are getting any hints that it can be a drug lead, then we are stretching that things to our wet lab so that we can work on that. And this is a view of our wet lab. So I will shortly describe you how our lab is looking like, because some people would prefer to come after your selection to our lab. So I just want to show you the facilities available here. And see, this is we have the analytical laboratory, and for the, your HPLC, HPTLC, that analytical things you want to do. So we have a very good facilities here. We have the wet lab here where you can prepare your media and the biochemical experiments. And we have also a phytochemical lab with a good fume hood, and so many facilities are here. And that is the molecular and cell biology lab. And this is, we have 3 biosept cabinets actually, and I showed you here one, because one we are using for particularly the bacteria and actinomycetes, and one you are using for specifically for fungus. As you know, the fungus having the spores, it can make some contaminations. So we have separate biosafety cabinet for fungus, and we also have another biosafety cabinet in molecular biology lab. That because that things are very sophisticated. So overall we have 3 biosafety cabinets, and this is our microbiology lab. So, and this room is our cold room. We have a very good cold room so that we can store our cultures, and those who are water we are collecting from the shodhyatras and different that soils and water and anything. So we have this cold room facilities to maintain our cultures. And this is the sophisticated instrumentation we are having. Just like this is the atomic absorption spectroscopy. And I think you people, now your curriculum is very strong. So this is, there was a time in undergraduate level, we don't know about, we never heard about these instruments, but though you don't know about the exact application of these things, but still you heard about this instrument. So this is atomic absorption spectroscopy. So what we are doing, the measuring some concentration of metallic elements in different materials as an analytical technique. And the next one, we have a very good HPTLC facilities, which we are using for purity control of chemicals, pesticides, steroids, and water analysis. And also we can use it for analysis of vitamins, water-soluble food dyes, pesticides in fruits, vegetables, and other stuffs. And next, this is the HPLC. And I think you people know what purpose it is used for, this HPLC. So I know in chromatography, we are simply using some TLC and column chromatography, but this is the, high performance means pressure is used here. So it is not a, directly we can't go to take the crude extract. So before taking to the HPLC, we are purifying some different processes, our crude extracts. Then we are isolating, the pure compounds here that can be taken for identification, I mean structure elucidation and confirmation of that compound. And this is one gas chromatography and mass spectrometry we are having, which can be used for especially for volatile compounds, plant-based compounds. And we are having 2 centrifuges, one for small centrifuge and one big cooling centrifuge. We have a fermenter unit so that we can do the large scale cultures once we are getting any novel bacteria or fungi bacteria which is showing very good effects in any field. So we can do the large scale fermentation. And we are also having in our molecular biology lab the gel dock system, which can be used for gel imaging to record and measure the level nucleic acid and protein of various types of media. And also we are having the ELISA plate, that microplate reader. And this is the BOD incubators we are having, and orbital shakers. So this is used for maintaining our cultures for their growth. And also we are having the orbital shakers. And this is a lyophilizer. We have actually two lyophilizer. Here is one, and this one is another one. So we have two lyophilizer to, for what purpose the lyophilizer is used. So this is used to dry the water, because when you are extracting any compound, we are using different polar solvents. But methanol, ethanol, that is fine. We can, use the, and we also have the rotary evaporator. However, that is used for the organic solvents like methanol and ethanol, which can be dried there. But when it is a matter of water, it takes a long time to dry. So we are using here, it is a pure butter extract. So we are using the lyophilizer. So it can, freeze dry that things, and you can get your sample within short period of time. Then this is the one, details of our microbial cultures. So while during Shodhyatra, we have collected many soil samples from different hotspots, and we isolated this bacteria, fungi, and actinomycetes. We just categorized because, and some of you might be the microbiology students, and basic microbiology knowledge you are having. So we have bacteria, fungi, and actinomycetes. So first we are doing isolating that microbes in some media. We using some media. Then we are seeing, because we are using three different medias. For bacteria, first of all, we are using the nutrient agar media, and for fungi, we are using the PDA, and actinomycetes. There is specific actinomycetes isolating agar, or we are using the ISP2 media, so that those bacteria, the respective microbes will go to the respective media. And after that, we are doing try to get the pure cultures. Then we are just coding them as from which area it is collected, and what is that, whether it is bacteria, whether it is fungi. We are doing the morphological analysis. So after a long term analysis, we got these numbers. So finally we have 4,254 around. Now we are also on the process of developing more, so some more may be added shortly. So currently, as per our record, we are having 4,254 number of bacteria, 1,923 of fungi, and 2,613 number of actinomycetes. So overall, we have around 8,790 numbers of microbial culture with us. And we are just doing this type of program that you people take interest, and the resources are with us. It is our duty how to explore these resources, because we know that so many drugs you read in literature, so many drugs in the market is now from the bacteria, whether it is from bacteria, whether it is from actinomycetes, and this is very natural. So why don't we try that? We have a lot of cultures, and we will do some experiment on that, and we will apply that products in the agriculture, or in veterinary, or in human health, and anything. So we can do. So you will be very happy. You don't need to go to field to again collect some sample and again do the process. The already samples are with us. You come and do your experiment. And one more information I would like to tell you, because don't be panic if you are not selected, because this is a competition. So we are going to select 10 students. That doesn't mean other students are valueless. They are also same genius like the selected students. So if you are really interested to do your work or do your research, our lab is open for you as well. You just simply, but your entry should be in a different process. Like that, you just give us one recommendation letter from your supervisor, wherever you study, that I am from this institute, and I participated in your BIIS workshop. However, I was not selected, but anyway, I'm really interested to do some research. you can come for 3 months, 4 months, and you come and you work on these microbes. Okay, so that is also open to you. And next is, I would like to say some of the ongoing activities of our lab. So this is the antimicrobial screening activities. This is a primary screening we are doing by cross perpendicular streaking. And after that, after getting some good results, that good results we are doing in well diffusion method. And we already got from our SMB, that's History Microbial Bank. These are the results showing the very good zone of inhibition activities, and they are also under process to extract the compounds which are responsible for showing this good antibiotic activity. And we are also, as told you earlier, we are also working on the agricultural things. So, you know, the phosphate things are, many phosphates are in insoluble form in the soil, but but plant can take that insoluble form. So it needs that something will be added to that. So that will be available in soluble form, and it will help the plant to grow. So finally, it can be a plant growth promoter. So we are doing some activities that phosphate solubilizing. You can see the good hollow zone here, which describes that our microbe is very effective in solubilizing the bacteria. But our next, we are confirming in our secondary screening by doing some biochemical tests, some indole acetic acid assays. And after the 2, 3, 4 confirmations, when we think that really it is working good, so we are doing the crop pot treatment for that. Then we will definitely take it to the field. And this is another assay that some cellulose degrading bacteria and some enzyme producing bacteria. You can also see this bacteria is showing a very good cellulose degrading activity. And also these bacteria are some of the recognized bacteria of our SMB, which is showing some good enzyme producing activity. And also we are working on the pigment producing thing, and you people know how pigment matters to us. Anywhere you go, we need colors for food coloring, we need colors for dye coloring, dyeing the clothes, and we also want colors for, you know, our making the tablets and capsules. Also we need color. But however, you imagine if we are using synthetic colors, how harmful it will be. So this is one attempt that we will produce the natural pigments from our bacteria, and we will isolate that pigments, and after isolating we will see the toxicity levels, and we will do in different applications. So it might be food grade application, it might be, you know, different, and in the cosmetic market also, you know how color matters. So we are also going to work on that. This is some glimpse of work that this type of pigments we are getting. And this is another experiment, sorry, like, just 1 minute. This is a seed germination thing. So we are using some weed extracts, you know. Usually we know the weeds are valueless, and we are simply cutting and throw them off. But we tried some 5 to 10 different weeds, and we took them in different concentration by making the extracts, and we tried to germinate the seeds, and we observed how fast the seeds are growing. So we got very good results, and the pot experiment in progress, and probably within 1 or 2 months we will get that results, and we are getting very successful rates in that, and we are happy that definitely in a short time we will get one more agricultural product on that. And next is we are also working on some entomopathogenic fungi that things we are also have a separate lectures on these. This is a really interesting work done by our scientist, Dr. P. Vivekanandan. He will give you a lecture later on this workshop, that how he isolated these things, this fungi, and this fungi is itself used in as a pesticide, and he also developed one product on that. After my session, he will take the session, and he will describe you the whole lab products we developed from our lab. So it will be very interesting for you to know that by using the traditional practices only, how we are successful in developing products. Okay. And we also tried one to use some develop some low cost media, just like the media we are using to isolate our microbes. It is costly. So we are also trying that what nitrogen and carbon sources we can give. So we just attempted that we use some black gram, finger millets, and simple the soil itself we added with agar, and we tried to isolate some of the microbes. So this is the first phase thing. So after that, few days we found a lot of microbes on that, and I showed you in my last slide that one halogen in cellulose degrading. This was from this bacteria only. So that is also an attempt that not only we will only dependent on the chemical farms for the media, we can also develop our own media, and we are also working on that, so that we will try to make some low cost media, and we can make it self life so that it can be used in our in any laboratory. And this is also one of the glimpse of the ongoing activity, that we are making this paper based device. Now it is in the product form, but however it is ongoing every day. So few research persons are working on this, that how can it will be easy to take your blood group on your own. So we are also working on this device, and also one mastitis device. Okay, thank you. And I think if you have any questions regarding our lab, because think positively that you are going to selected, or anyway, I told you this lab is open to you. So you can come here anytime and work. So regarding these activities or any queries you have, you please ask, and I will be happy to answer you.
Students, if you have any doubts, you can ask Dr. Premlata.
Good morning, ma'am.
Good morning.
I have a query about the Shodh Yatras. So are they still happening?
Actually, since last 2 years, the global situation, the nationwide situation,
Yes,
because
yes.
there is not allowing of any gathering. So that, because usually I just forgot to show you some photos regarding our Shodh Yatra. It is with some 60 or 100 people are at a time are going. So once the pandemic activities will be very weak and we will recover fully, that we will be allowed, that we will not take any permission like that for that gathering, we will again start this.
Okay, ma'am. And one more query. So the like regarding the past Shodh Yatras. So like, because I think from there only we isolated even the microbes, right, along with the traditional rural practices.
Yes.
So like I think you from the yatras also, you kind of like could pick up some microbes, right? So so which all areas did you like cover, and what was the soil profile in those places?
Yes, actually, as I told you, that we are selecting some before starting the Shodh Yatra, collecting the soil sample, because every area has its own importance. Just like some areas are having a lot of pollution and other things. So we just think that this area might be full of that. What is that, heavy metals or something, but we try to isolate the soil sample of that region, so that we will get some bacteria which is resistant to that heavy metals, right? So like this, we are choosing some hotspots, like we are also going to some associated marine habitats, and we are also going to as I told you, the extreme environment areas. Just imagine in a heavy winter we are going to Jammu Kashmir, so that area is full of covered with snow. From that area we are trying to isolate the soils, because there is a logic that in an environment, in a harsh environment, any microbe or any organism, they're adapted to that environment. Why? Because they have some chemical reaction or chemical strategies in them so that they can adapt. And we are getting that cues that if that mechanism is in that organism, why it is not, why it won't be helpful in combating any disease or any problems in outside that environment, right? So that's why we are choosing the areas that may be called as some extreme environment areas and some harvested areas. So different, that's why the in whole India says a north, south, east, everywhere they're going and they're collecting samples so that we could know that after getting some results, we could able to make a very broad idea that this area sample is showing this type of result, right?
Yeah. Thank you, ma'am.
And I want to ask you people some question. While you submitted this application form for this BIIS program, you definitely gone through the guidelines, and what is your expectations from this workshop? Anybody?
Good morning, ma'am.
Yeah, good morning.
So, ma'am, I'm hoping that I could learn about this industry more and I could learn new skills and knowledge which I would be able to apply in coming years. So that is my aim for joining this.
So, Arushi, you are in a BSc student or MSc?
BSc, I'm a first year.
First year BSc. Perfect. Yeah, because see, undergraduation is a situation where actually nowadays this is, I would say, this is the first platform that which is promoting the undergraduate students to do research, because once we are in the MSc or MPharm, or once our master started, we are in our every lecture of our professors, we are every day we are listening about the research, right? But in an undergraduate level it is quite difficult because we have our own curriculum and we are just on into that curriculum, we are not going outwards. My opinion is this BIIS workshop is such a platform which will help you. It is your first step in research, I would say. If you are, some of you will be go to any much more higher studies, but you will remember this workshop is your first step, because this is the first week we just designed, we are giving lectures and overall ideas, but later on in the next coming 2 weeks you will learn the subject things, then you will able to think that okay, this is the thing, so why I will be not a part of that, why I will not do the research on that. So that's why my sincere request to you people that listen each lecture sincerely, and so that we will really extract that 10 students which are the 10 student we are not selecting like that, we will be very satisfied if that 10 students after their completion of research, that things will come in a 10 products. So from today itself, be serious that this workshop, we will not take it so lightly, we will take it as a challenge. Okay, so we are now, you are now 65 to 67 students. Now competition is very strong. You just make a challenge that I will be the winner. Okay, and I will do the project, I will do the traditional practices, and I will be the part of that product. Okay, so listen carefully. If anything is, any doubt is coming, so you have the WhatsApp group, you can ask there. Any lecture is going on, so there are a lot of, you will enjoy the sessions of many eminent professors of all over India we collected this time. And usually probably each year we are doing, but this time we will learn a lot from them. So they personally, you can interact with them. Just say you are going to do some more research, but you need a reference. in your workshop, if you recognize, okay, that professor was from that Bangalore, that IISc Bangalore. So I can put a word in my email, sir, I just heard your lecture in your BIIS workshop. So I just impressed with your lecture. Now I'm interested to do this research. Could you please help me? So just imagine how that email will impact to them and impact to you as well. Okay. So now this session I am ending with this, and the next session will be conducted by Dr. P. Vivekanandan. He will give you the he will give the details about that products of our Srishti lab, and you enjoy that session. Okay, thank you.
Thank you so much, Dr. Premlata. The next session will be by Dr. Vivekanandan. He is an entomologist and also a scientist at Srishti Laboratories. He will give you a glimpse of the products developed through various grassroots innovations, which are worked by students like you and also other scientists. Okay, so let's welcome Dr. Vivekanandan.
You are muted.
அதெல்லாம் சைடுல இருக்கு. Slide move on. Okay. Very good morning, Dr. Cynthia. Myself, Dr. Vivekanandan, scientist at SISTI. Today, I talk about SISTI product development. The SISTI product, so main aim is eco-friendly product for agriculture, veterinary, and human product without chemicals. A chemical-free product. In previous session, Dr. Premata Madam explained all the aim and objective of the SISTI and the product. SISTI have developed several agro products for growth promoter as well as biopesticides for crop protection. SISTI have developed several agro products. And for example, the SISTI Saxi is a growth promoter, and SISTI Prias, the growth promoter, as well as the reduce the flower setting, and SISTI Sakti growth promoter, pest reduction in cotton plants. SISTI Suraksha growth promoter and protection against leaf curly diseases. SISTI Suraksha is a growth promoter to enhance the growth of the plants and also control the termites. And SISTI Praga, the growth promoter, protection against mealybug. SISTI Sarvatra is a growth promoter, protection against shucking pest. And SISTI Sudarshan is a growth promoter, protecting against chewing pest. And SISTI K and C, the growth promoter, protection against plant pathogen like bacteria and fungi. And SISTI Shara, it's a growth promoter, pest control during the flowering of fruits. And also SISTI have developed several animal products for cure the mastitis diseases, diarrhea, intestinal worms, and cough and cold, and some other diseases in animals. The musty herbs cure the mastitis in animal, and hepatic herbal hepatizer rip herb herbal product to cure the cough and cold in animals. And worming herb is a herbal product, the botanical derived product to control the intestinal worm. And the bloat herbs, herbal product to treatment of bloat diseases in animal. Bloat means the product is a herbal derived product for the treatment of bloat diseases in animal. Placenta case, the herbal product to easily relieve the placentas in animal. Diarems, the herbal product to control the diarrhea. And a heart herb, herbal product to cure the arthritis. Heart herb, the herbal product to cure the arthritis. And galactogogue, the milk enhancer in animal. Milkomax, the milk enhancer in animals. Utriocare, herbal product for the uterus. And rem, herbal product to wound treatments. SISTI has developed several human product for pain relief and skin care product and mosquito repellents. And the mosquito hits is a mosquito plant derived mosquito repellents. We know the mosquitoes are small insect pests that can carry the very dangerous vector-borne diseases like malaria, dengue, chikungunya, yellow fever, Zika virus, so many very dangerous diseases. So SISTI has developed mosquito hit for mosquito repellents of adult mosquito. Herbo heal, wound healing ointments. Herbo glow, antiseptic skin creams. SISTI pain relief, natural pain reliever. Care and cure ointment to heal the crack and other good ointments. And Zumate, eczema and psoriasis, this control the human diseases. This product was licensed by FDA. We have developed a paper-based device for self-checking the blood grouping. Like within the 5 minutes, we will checking self blood grouping. Okay. Another one is a paper-based mastitis detection kits. We will check the mastitis diseases in animals in blood. We will easily identify it by using the 60 paper-based device. We also focus the fungal derived product for agriculture purpose. We have isolated several biocontrol fungus, like Trichoderma species. We have isolated several biocontrol fungal species from healthy plant rhizosphere soil condition. First picture shows we have isolated several biocontrol fungi, Trichoderma species. Among the biocontrol fungi, Trichoderma species, we have screened Trichoderma azarnium, more potent biocontrol fungus against plant pathogenic fungi and bacteria, as well as this particular Trichoderma azarnium can control the small insect pests like happy and some other small insect pests. This this picture shows first two pictures shows before treatment of Trichoderma azarnium, and that first two pictures highly affected by a small insect as well as the plant pathogenic microbes, bacteria and fungi. Third picture showed after treatment of Trichoderma azarnium fungi. After treatment, there is no microbial, the bacterial and fungal infection was observed, not observed, as well as there is no small insect pest was observed after treatment of Trichoderma azarnium. Among the all Trichoderma species, this Trichoderma azarnium has more potent activity against plant pathogens as well as small insect pests. We also focus entomopathogenic fungi. What is entomopathogenic fungi? The entomopathogens means only pathogenic to insect, not other pest or species. Only targets pathogen to insects. So we for pesticidal purpose, we have isolated several entomopathogenic fungi such as the Bavaria bassiana, Metarhizium anisophilae, Verticillium lecani, and Escherichia species for pesticide purpose. This first picture is nothing but Escherichia fumoseraceae, the fungal spores formulations. Second picture is nothing but the before treatment of lemon caterpillar larvae, 4th instar larvae of lemon caterpillar larvae. Third picture is showed after treatment of Escherichia fumoseraceae fungi. This particular Escherichia fumoseraceae fungal spore cause 100% mortality rate after the 7 to 10 days post treatments. And also you visibly seen the fungal growth was observed on dead insect cadaver. This microbial pesticides has several advantages compared to synthetic chemical insecticide. Once we use the synthetic chemical insecticide for control the insect pest, the chemical pesticide cause several side effects to ecosystem and other non-target species. This microbial mediated the pesticide is target specific without pollution, is a cheaper compared to synthetic chemicals. It is a eco-friendly method for control the insect pest. And this laboratory and field evaluation of entomopathogenic fungi. The first set of a picture showed laboratory evaluation of entomopathogenic fungi on different insect pests. Second set of pictures showed the entomopathogenic fungal spores formulation was tested again in field conditions. Based on the laboratory and field result, we formulated the two set of fungi, the biocontrol and the entomopathogenic fungi. They formulated with talcum based powders. It's 98.5% carrier, the talcum based powder formulation. After once we formulated, we supplied to the farmer for his feedback. Based on the farmer feedback, we have developed the final product for commercialization purpose. This work was done by previous BIIS students. This study shows good results. So we further we will focus for further product developments. The first project title is isolation of chitinase enzymes from chitinase producing microbes. Microbes from the western coastal regions of Gujarat. First, we collected shrimp shell waste and washed with running tap water to remove the unwanted debris and particles. After that, we did demineralization by soaking in 36% HCl, hydrochloric acid, for 1 hour. After that, we washed with running tap water. Then we performed deproteinization by boiling with 3% of NaOH for 30 minutes. After that, washed with running tap water and dried in sunlight. After that, we powdered the shell and make it chitin. This chitin was used for isolation of preparation of media, chitin based media. Chitin based media very useful for isolation of chitinase producing enzymes. Okay, this is why we want to use this particular chitinase for agriculture purpose. The most of the insect and plant pathogenic fungi cell wall is made up of chitin. So these chitinase enzymes involved for lysis and rupture the insect and pathogenic fungi cell walls. This will highly effective biopesticides for pesticides in agriculture. So we focus the chitinase enzyme. So after that, we isolated the chitinase enzymes from chitinase producing microbes. After that, we tested different insect pest as well as plant pathogenic microbes. Second, evaluation of grassroots practice for crop protection. Okay, it's a Azardiga indica neem fruit extract with combination with cow urine was tested on Hapid and agricultural pest control. This particular formulation highly effective on control the aphids in brinjal plant. These results clearly show this formulation highly effective in after 3 days post treatments. These formulations, this grassroots formulation was tested against major insect pest. This formulation was agave americana leaf extract was tested on different insect pests, mealybug, heliothis, and different insect pests. This formulation was highly effective in after 7 days post treatments. These 3 product was we plan to develop as a product further. We have filed 12 patent, more than 12 patent, and granted 3 patent. 60 has got several award and recognition, and 60 has developed 66 product in agriculture, veterinary, and human, and 7 products as under developments. And this is a publication. We have published several national and international publications in reputed journals. These are a publication. Thank you. The 60 main theme is chemical-free solutions without chemicals. We know the chemical cause several effect to non-target organism as well as the green ecosystem. So we develop only eco-friendly solution. Okay, in the field of agriculture, veterinary, and human. Okay. Okay, thank you. You have any doubt? Please ask. Student?
Students, if you have any questions, you can ask. If you have no questions, then we will wind up this session. And after 10 minutes, we will once again come back together for the next topic. Now we have another 2 lectures today. One is Dr. Vanita Prasad. She is an entrepreneur. And then we have another session with Professor Anil K. Gupta. He will interact with you on documenting the practices. Okay, so stay online. students. Professor Anil Gupta is with us, and Dr. Vanita Prasad is also with us.
Good morning, friends. How is everything going on? How are these sessions going on? Are you able to enjoy, absorb, learn? Any quick reaction to the sessions so far before I start?
Morning, we had a good session with Dr. Taslim Arif from CCAM.
Very
Yes,
nice.
very good questions also students raised and had a very good interactions.
Very good. Very good.
And then we
Anybody
had
else has any comment, any observation, any suggestion how to make it even more useful than what we are trying? Any student wants to say something, please switch on your videos. Please switch on your videos. Switch on, Anjali, Deepa, Neha, Kenja, Sanyukta, Arushi, Rohan. Please switch on your videos. Sindhu. Shouldn't take so long. If you're all around, then it should be just a matter of few seconds, you know. Neha, Kenja, where are you? Vaishnavi, Sanjana, Ria? Are you there, or we will remove you if you are not going to switch on? Please understand that. I'm not able to understand. Why should it take so long? Sanjana, Sanyukta, Pratyusha, Ria, Tejaswini, Thipa. What do we do? Riha, you're not there, then shall we remove you? Arushi, you're also not there? We'll have to remove everybody who is not there. Please understand. This is the last chance for you to switch on. Either you speak up, unmute and tell us if there's a difficulty, or we assume that you're not there.
Sir, I have
Sakshi?
on my video. It's like the same.
Sakshi, okay, all right. Sakshi is all right. You have replied. Anybody else who has a difficulty, please chat, put a chat.
Sir, Amulya here. Sir,
Yes.
I have on the video, but the camera is disabled. It's
All
covered.
right, all right. Who is it?
Amulya.
Amulya, all right, that's fine. It's okay, so you can switch off your video, then there is no problem. Yes. Arushi, Anjali.
Sir, my video is on. Is it not visible?
Anjali, आपका नहीं है वीडियो बच्चे on.
Okay, sir, I will rejoin.
हाँ, rejoin कर लो. Arushi, Thipa, Peryar. Thipa, I'm removing you now. I have given you enough chance. You are being removed. Okay. Sanyukta, you're also not responding. So I'm removing you, Sanyukta. Remove. Arushi, you're not responding. Arushi, unmute yourself if you're there. Arushi, please unmute if you're there. No, so remove her. Where is Arushi? Arushi को remove किया? I'm not finding her. Maybe she has. Yeah. So I will remove her. Okay. Then what about Dhruv and Ria? Dhruv is okay there. Sindhu? Ria? Ria, you're not there? Last chance. Okay, removed. Sanjana? Pratyusha? Are you there? Vaishnavi? Pratyusha, you're not responding, so I'm removing you. And Sanjana, what about you? No response. Please keep checking, huh? If somebody is not there, we don't report, but we will not let them keep this on. I mean, we would like to have people who are interested in attending.
Okay.
Vaishnavi? Vaishnavi? Vaishnavi, you're not responding. Okay. Removed. Shubhangi? Shubhangi?
Yes, sir.
So you should respond. You cannot switch on your video. Shubhangi? Yes. Okay, but इतनी देर से मैं बोल रहा हूँ, आपको नहीं सुनाई दे रहा? You are not able to hear? I've been asking for last few minutes.
Sir, I am
Thipa?
having network issue.
Okay, then you can switch off. Thipa?
Sir, now only I've joined, sir.
You just joined now. All right. So we have now 24 students, which is wonderful. We like to interact with you all. And Amulya has already been informed. Yes, Amulya has a problem. That's okay. Sindhu ka kya hua? Sindhu replied. Sindhu, are you there? Sindhu? No, Sindhu is not there. So removed. Okay. Friends, what we're going to do now is to discuss something very important, which is about how do we scout and document innovations, traditional knowledge, and other creative solutions that people produce at the community level. And why do we want to find them out? Because over the years, sometimes decades, sometimes centuries, sometimes millennia, when the modern means were not available, people had to discover local solutions to cope with the complexities of life, to deal with risk and uncertainties, to look at climatic fluctuations. So they had to find some solutions. And during this search, sometimes we have discovered over the years, last 4 decades, 35 years ago when Honeybee Network started, and our research started even before that in 79, 80, that many times some of these solutions that people developed themselves could extend the frontiers of science, could reveal new insights about problem solving, new heuristics. That means new ways of thinking, new ways of problem solving, about which little bit I'll talk to you about. So it is obvious that we want to learn both from traditional knowledge that our elders have. Did you collect some knowledge? The assignment that I had given, has anybody collected already something? Any one of you has documented the knowledge of your elders? I had given you 2 assignments. One, to document the knowledge, distinctive knowledge from your elders. And second was from, what was the second assignment? Tarumay, what was the second assignment?
So we have to speak to one of the service providers.
Service provider, correct.
Yes.
Yes, you are right. You are absolutely right. So who would like to answer, share something if you have already done that assignment? Has anybody pursued this subject already? Kanza, Tejaswini, Saurabh, Mohan, anybody has done this?
Excuse me, sir. Good morning.
Yes.
Saurabh. So I have just asked my grandmother something about traditional knowledge, because in our house, whenever we have pain, ache in our leg or hand, we use red sandalwood. We make a paste of red sandalwood and apply, boil it mildly, and then apply over that area. And the pain is relieved within 2 to 3 hours.
3 hours, okay.
Yeah.
So you don't rub it, you boil it.
Yes, we mix it with water some. We grind it on a stone.
Okay.
We make it a liquid type solution.
Like a paste.
Then we boil it mildly and then apply the paste over that area.
Okay. That's very interesting. I have not heard about boiling part of it. Application part I had heard, but not the boiling part. That's very interesting. Who told you this? Who, your grandmother or grandfather?
My grandmother. We practice.
What is the name of respected grandmother?
Rajini Pawar.
ऐसा नहीं बोलते ना? Mrs. या श्रीमती रजनीकांत है ना? दादी है, नानी है तुम्हारी ना? With respect, please take the name of elders with respect, है ना? Okay. So you should either put it on the chat or send a mail to us, so that will be very useful for us, all of us. And we have so many students, some are unfortunately absent today, but every one of you who is present here can add to this pool. So all of us will get, say, 30, 35 or 40 solutions. And this time, as Amar has already explained to you, we are going to get you back at the lab, isn't it? For a week, you're going to come here and do practical, operational experiments in the lab. So we are looking forward to meet you in person also. And in some cases, we will let you experiment on the solutions that you have documented. Wouldn't that be nice? That you also do an experiment on something that you have learned from the elders or from the service provider, and tell them that, look, we tested your idea. At least preliminary studies show that it might have real scientific basis. So your respect for their knowledge, the elders' knowledge, will increase. At the same time, they will feel good that you didn't just listen to it and forgot about it, but you have pursued further investigation. Imagine if yesterday I was talking to the secretary, DSIR and DG CSIR, and they give, CSIR gives fellowship to the students, even from non-CSIR institutes. About 8,000 students, doctoral students, get fellowship every year. Now, knowledge of the elders. So please understand that this is a knowledge which is under threat. Why are we doing scouting and documentation? Because this knowledge, it is getting eroded. So just as we make a dam to prevent water and soil erosion to take place, we make a dam, or we make check dams, small check dams to store water and also to conserve soil. Same way, when knowledge is getting eroded, knowledge is getting lost, knowledge is getting forgotten, we need to document it. And for you, when you become grandparents, understand, Neha and Kenja and Tejaswini, when you become grandparents, your grandchildren would have access to this knowledge. So 100-year-old knowledge, we are trying to make sure, Honeybee Network and Trustee and Gyan are trying to make sure that this knowledge is available for next 100 years. You understand? So that we at least do our bit. We would not let this knowledge be lost. Every time an elderly person dies, is buried or cremated, a library that day is disposed of. Correct? All the life's experiences, experiments they have done is lost that day. Can we afford to lose it? No, we cannot afford to lose it. Therefore, documentation both of traditional knowledge and contemporary creativity. Contemporary creativity means some problem you faced last month or last week, and you try to find solution. Or you discovered some traditional knowledge and you made a change in that, either in the ingredients or in the process. For example, just now when Saurabh mentioned about red centers used in relieving pain, we had heard about paste. I had never heard about boiling it. That was a new addition. So some knowledge that I had now has been invigorated. It has been valorized by what I learned from Saurabh's grandmother. Correct? So we are also, there are many teachers in this room who we have not met, but someday we will request Saurabh, please let her join our class online, and we will thank her. The grandmother, and we will, like all your grandmothers and grandparents, we will have in a class. It's no problem. Has it ever happened that your grandmother has come to the class? आएंगी ना, इस कोर्स में हम बुलाएंगे उनको. We will invite them. All of you will get a chance to invite the elders as well as service providers from whom you have learned, and we will give a collective ovation to them that look, you are all respected colleagues, are our teachers, and we want to salute you and thank you that you have given us knowledge produced in the laboratories of life, as Dr. Mashelkar often says, not just in biotech laboratory or natural product laboratory, but also laboratories of life. And mind you, they love us so much. So obviously, when they try something, they will try it after a lot of experimentation and testing, isn't it? They will not tell us something which is not safe. They will not tell us something that they don't believe in or they have not tried. So it is a tried and tested knowledge. Only thing is, they may not know the biochemistry of it. They may not know the genetic basis of it, if there's any. They may not know the biochemical or other basis of it. That's possible. But that's a language issue. That's an explanatory issue. That's what we are supposed to do in BIIS. We are training you and building your capacity so that you can explain these things better. Right? Anybody else wants to share something before we proceed further? Ayush has mentioned use of Kataka seed, Strychnos potatorum for water purification. How do you use it, Ayush? Can you explain? You can do an experiment on this right away.
Yeah, so my father said that I spent my entire childhood in Koraput of Odisha, which is a very backward district in Odisha.
We have done a show, the Yatra in Koraput.
Yeah,
so I know that place, yes.
Yeah, so he said that he has observed tribals in that area using like grounded dried seeds of Kataka as like natural coagulant and flocculant for water purification.
Wonderful. So now, what is your father's name, please?
Shri Sibasis Panda.
Panda. What does he do?
He is a chemist, like analytical chemist in waterworks.
Did you find out what is there in his Strychnos which does this? Have you tried to search it on the web? Not yet.
Yeah, I did look up. Like I think I'm not sure, but it has negative charge, so it kind of like attracts the opposite charged dust particles, and the turbidity goes down or something like that.
Let us find out. This is something, seeds of Strychnos potatorum, natural water treatment. Now there is a paper on this, Bache. And this January 2014, Pakia Lakshmi published a paper on screening the water quality assessment of drinking water using the seed. And let me put this paper on the chat so that you can all read this paper. So you see, you can tell your father and your family that, look, we got a full text of this paper, and the research was done to find efficiencies of powdered seed for water treatment. And this is a study from Tamil Nadu. And Pakia Lakshmi, Suganya, and V. Guru. Let me share this briefly so that you know that this is what the purpose of us is, not just to celebrate this knowledge. Can you see this paper now? Can you see this?
Yes, sir.
Yes, sir.
Yes.
Yeah? Now, exactly on the point that you mentioned, isn't it?
Yes, sir.
What's the name you said? What's your name? Ayush?
Yes, sir.
Now, look, this is a paper published in 2014. All right. But the communities in Koraput may not have heard about it. Correct? That is why when we were discussing ethical issues, one of the principles we said in Honeybee Network, that whatever we learn from people, we should give them back with value addition. We should tell them a little more science of it. So what these researchers have done, they have seen that the plant can be used for the treatment of turbidity in drinking water as primary coagulant. Compared with alum, the seed extract is effective as a primary coagulant compared to alum. It produces water with slightly higher residual turbidity and residual color, but the residual turbidity and residual color are within the WHO drinking water guideline value. Turbidity 5 NTU and color 15 TCU. The total solid has been investigated. Preliminary phytochemical screening was carried out. Higher spectrum was also done. And their antibacterial properties against gram-negative and gram-positive bacteria were also seen. Growth of Proteus vulgaris, Pseudomonas, Staphylococcus epidermidis, E. coli, was significantly inhibited. So it is not merely doing coagulation as we were thinking, Ayush, it is also making water bacteria free. Correct? It is also killing the organisms. So maximum of, and some of those organisms which are infectious have also been corrected. This is a paper from Tiruchirappalli, Tamil Nadu, PG and Research Department of Microbiology. So now you see what are we trying to do in BIIS? We want you to equip with the skills so that we produce knowledge of this kind. And this would not take too much of day, too long an experiment. This can be done in 2-3 days. I would like our team in BIIS also to note this. Many of the practices we can validate in few days. They don't require months. And but only thing is, we should acknowledge the source of knowledge. We should mention from where have we learned this, correct? And that community should get an Odia translation, Ayush, of the findings in Odia language, isn't it?
Sir. Yes, sir.
So that the community also learns. Otherwise, what happens? We learn, we grow in our life, but community remains where it is. That's not fair. It is unethical, isn't it? Not proper. It becomes extractive. It becomes exploitative. So as you can see, they have this whole paper here. You can download. And then there is somebody who has done the medicinal uses of this plant. It is called Nirmali in many parts. Seed, bark, root are used for chronic diarrhea also. So mind you, when the diarrhea is one of this a consequence of impure water, isn't it? So not only that it purifies the water, it adds something to the water that if you have got infection from some other source, it will also help heal. So water becomes medicinal in some respect, isn't it? So this is the kind of documentation we want to do. We would like all of you, Saurabh, you will also have to do similar research on red sanders properties, you know. How does it, what is the anti-inflammatory property, correct? Saurabh, you understand?
Yes, sir, sure.
So then you have to tell your grandmother that, look, I did some research. I found that, yes, this knowledge has been known to come our country, our scholars, and they have found this, this, and this property. She may not understand the technical term. Explain to her, what is anti-inflammation? What happens when there is a pain? And tell her in simple language, in Marathi. You are from Marathi, you are Marathi speaking, isn't it?
Yes, sir.
So tell her in Marathi how the pain is reduced. Does it, what are the ways in which pain can be felt by our body? Anybody? Give me quickly, what are the ways, what are the pathways by which we feel the pain?
Sir, the release of prostaglandins.
One. Second? When we give some medicine for pain, what does it do? Does it have to necessarily remove the pain? What else can it do? It can do that. It can reduce the inflammation, right? What else can it do?
When we take painkillers, sir, it subdues the pain.
Or sometimes the pain is not reduced, but what happens?
So it
For the receptors of that
like
pain,
we do not feel that pain.
deactivated.
More, I want a scientific explanation. What happens when the pain is still there, pain is there in the body, but I have taken a particular medicine. What does it do?
Sir, the receptors.
Where?
Sir, the prostaglandins and the receptors, which are generally responsible for the inducing of pain, gets deactivated for some time.
When you are not, what is happening? Which pathway are we blocking when you have the pain but don't feel it? When the brain doesn't receive the signal, isn't it?
Nervous system.
Correct. But the pain is there. So sometimes the feeling of the pain is inhibited. So sometimes the feeling of the pain is gone. Correct? We have to help. We have to help our communities, our elders, our service providers get some idea who is this person. Please check 728516211. Don't allow unless you know who is this person. You understand?
Okay.
Ask that person to identify.
Good morning, sir. Good morning, sir. Sorry for the internet issues.
Vinita, we'll finish this. Can you give me half an hour more? Is it possible?
Yes, sir.
Then I can finish this part of the discussion and then you can take it. All right?
Sure.
Yeah, so around 12:30 maybe. Sorry, 12:30.
Okay, I will join at 12:30.
Yeah, please. Thank you so much.
Sorry for the
No problem. No, we understand. So we need to also go into the question of what this medicine, what it might be doing. Is it suppressing the inflammation? Is it reducing the inflammation? Is it removing the sensitivity of the inflammation? Is it that I'm not getting the signal of the pain? The pain is there. Or is it actually reducing it? Both possibilities are there. Anybody else wants to share anything before we proceed on methods of documentation? This is one method, of course. The one that we are discussing just now is a very empathetic method that we talk to the elders in our own families, in our communities. We go to the villages. Maybe during Shodhyatra, my colleague must have explained to you what is Shodhyatra. Chetan must have explained. We walk from village to village, stay in the villages, and share with them what we know and learn from them what they know. So there is a exchange of knowledge and we document it. There are many other methods I will mention in a minute. Anybody else wants to share something? Anybody else has to talk to the elders in your family?
Not to my elders, but then when I went to Chittorgarh in Rajasthan, they used the Sitafil quilt they told that they use the Sitafil quilt, they dry the outer covering of the Sitafil and then they make a quilt out of it to relieve joint pain. I don't know how it
Quilt out of quilt?
Yes,
Sitafal
sir.
meaning the custard apple or Sitafal meaning the gourd?
No, sir, the custard apple. They actually used to make saris in the olden days.
Saris
of custard apple.
Really?
It
I'm hearing for
originated
the
from
first time, Sanchi. You have to give me more. Do you have any photograph of this thing that they have?
No, sir, they didn't permit photograph, but they told that it originated from Padmavati's era. So she started this, and from that they had the idea of making quilts, and they told something it has magnesium. So that magnesium helps to reduce the acid in joints, and that relieves the pain. But that's all I know.
My goodness. Can you not get a photograph of that quilt or at least the process of how they get the fiber out of it?
So I can search on the internet. I'm sure they would have.
No. Okay, I can try because I have never heard this. This I'm hearing for the first. This is of the seed or of the coat?
So the coat.
Okay, custard apple. Sari made out of custard apple. You're right. You're very right. Oh my goodness. I didn't know that. And I never knew, but there are, let me see, custard apple is there, but can I find saris of custard apple? Yes. I'm not able to find custard apple, but there is certainly you find that on the net. But this I somehow did, I never knew about it. No, custard apple, sorry, we don't have it here. Blue custard apple, sorry. Maybe they are using the custard apple for the color, or I don't think this is made of custard apple. But we'll have to check. Okay. But yes, you custard apple by sari made of custard apple. They are in fact mentioning in Rajasthan. Oh my God, sure. In Chittorgarh, you're right. You mentioned about Chittorgarh, isn't it?
Yes, sir.
Okay. from fiber obtained from plant like bamboo, banana, cotton, custard apple, coconut husk, and Rajasthani rajai, lightweight blanket, medicated quilts. Okay, very interesting, very interesting. Now that's very interesting. I didn't know this, but that's very interesting. Check up Amar, who is 728051621. I don't understand why they have not
I
named it.
asked him to identify with his name, but still he is using the number. I'll check.
He's not done that?
Yeah, he has not done that.
Then we will not admit.
Okay.
Unless the person identifies and uses, we had mentioned very category. You please put in the group also that we had to remove.
Already put it in the group.
All right. So tell them that they will have to join with their names. They cannot join with a number.
Yes.
Name, their name and institution name. That is not why. I mean, we must have. Everybody has done that. Why shouldn't they do it?
Okay.
Tejaswini, you should also put name of the institution also. Somewhere, Jaslyn, you should also put, Anjali, you should also put name of the institution briefly so that we can identify. Most of you have done, some have not done. Please do that. Okay, coming back. So anybody else wants to share? This is very interesting what you shared, Sanchi. Now, the question is which kind of medicinal value it has and how is that medicinal value imparted to our body? So are there dyes in the clothes which when touch our skin through dermal route, some part of it is effused into our body? What is the process through which it happens? Belief may be there, but we have to find the scientific basis. So my suggestion will be that if you can get some small sample, then we can do the trial in the lab to find out whether the fiber actually has. Of course, custard seed extract, as some of you might know, is a pesticide. And it is also used for removing what you call these worms in the head. Kya bolte hain usko, juko? You know, you have the women generally have in the hairs. Kya bolte hain? Lice. Lice. You're right. So custard apple seed extract or powder is also used to clean our hairs of the lice. Lice can be there in girls or boys, anybody, but nevertheless, it can be used. It is one of the well-known traditional methods. So we understand that when you apply it, it does the insecticidal property. But how does that effuse or diffuse from the clothes? I have no idea, but it will be useful to know that. Okay, we'll have to do some experiments to see whether with perspiration, does something comes out of the fiber, which then has antibacterial or other properties or some other chemicals are coming out. We'll have to find out that. Now, question is that as a scientist, we do not want to believe something and then be quiet with it. No, that is why our society has not progressed as much as we should have. The BIIS does not believe in folklore unless and until they can find the science underlying them. Correct? We are skeptics. We are respectful, but we are skeptics. Our first reaction is we will try to search and find whether somebody has already worked on it. If they have worked on it, we would like to see if somebody has found the chemistry of it or molecular basis of that. And then we will see whether there are some questions which still remain to be answered. Okay, so please remember that the Neha has mentioned about ginger. Yes, that is well known. Now, what in ginger does this? I take fenugreek seed soaked in the water every day in the morning. What does fenugreek seed do to my body? Do you know that? What are the compounds released in the water after soaking which help my body? Nahin malum hai na? Please promise to yourself, each one of you, if you notice such practices in your family, you will try to find signs of them. Okay, otherwise our society will never grow. We will not become scientific. We will not have scientific temper. We will not have curiosity to find science. And if we find science, we might be able to improve the process. With less quantity, we can have better effect. We can find better ways of delivering it. Maybe cold water may not be as good as hot water. We will discuss that in the session on phytochemical extraction. That many times it makes a difference whether you extract it in water, hot or cold, whether you soak it and then extract it, whether you soak it for 5 days and then extract, or soak it overnight, then extract. or you just take the fresh one. All of these things make a difference to the compound that are released, generated in the water. Understand? So it's very important that when we observe these things, we'll try to study. Okay. Now let us go into now some of the examples that you have shared are very useful. And now we will look at the next step, which is what are the other methods of documentation. So one method, of course, is competition. So sometime we organize competition among the people, among children, among students, among farmers, among artisans, and mention that if you have any way in which you have done something interesting, send it to us. We'll try to do the prior art search. What is prior art search? The literature available, the patents available, the products available in the market, or practices available in the market. All of these are prior art. Prior art means knowledge already available, whether in public domain or in paid databases. But the knowledge which is available before we have started our investigation is called prior art. Correct? Clear? Now, prior art, what does it do when we find the prior art? What will be our learning? What we will learn? If we do the prior art search, what will we find out? Which attribute will we find out of the knowledge? Jaslyn, what will we find out? Anjali, when you do prior art search, what will you learn out of it? Tejaswini, what will happen? What will you find out? You have got a practice, you documented a practice, now you're doing prior art search. After that, what will you find out?
The scientific basis of that practice.
Or, that is one aspect. What else will you find out? Whether the knowledge that we have documented is novel or is similar to what is available in literature or in patents or on the web or in market or in the patent database or in the knowledge database. It could be in any medium, printed, visual, or audio, or text. It could be in different databases. It could be in the books. It could be in the journals. Any source in which knowledge is cataloged, we will find out, access those sources to find out whether knowledge that we have documented is novel. Sometimes knowledge may be old, but the method through which you validate it may be new. Or knowledge is old, but you can modify it to make it more effective. For example, method of using sanders as a paste was known. People use it on forehead and all of that, but we didn't know that you can use it as boiled. You can boil the extract and get that water treated on your leg or hand, which I'm going to try, of course, because I have pain in my right leg. And Saurabh, I will test it and tell you so that you can tell your grandmother. If it helps me, I will definitely give you feedback. So we will do prior art search to find novelty. And novelty could be in how many ways? There are 4 ways in which novelty can be there. Let me just show you that. Let me share a screen and I will tell you how would you find novelty. Okay, so there are 4 dimensions. I will take 3 dimensions: method, material, application, and business model. 4th dimension: old, new, old, new, old, new. Right? So all you can have old method, old material, old application, but business model can be new. So where taxis were hired before, the method was known, the material, taxi, car, well known for hiring, but they were there. application for traveling, transportation from one place to another. That was old. What was new in aware? The business model. Correct? That you could aggregate different taxi providers on a common platform so that you don't care whether which driver is coming. You only know that so agency doesn't own these cars. Agency doesn't employ these drivers. But they join this platform because they want to offer their services at the terms at which this company has provided. So they get more business. So the drivers get more money, more job, more visits. Customers get assured supply. So I don't have to try now. Taxi wala, who is next door. I will go to taxi stand. There's no taxi. I'll wait for the taxi to come. Not anymore. From 2 kilometers, 3 kilometers away, the taxi says, I'll take 5 minutes or 7 minutes or 10 minutes and I'll come. And then you are happy. At least after 10 minutes, you know that the solution is coming. So business model is new. Take the azadirachtin. So let's say we documented that neem is used as pesticide, correct? Neem leaves. All of you know that. And one compound which is used, there are many more compounds in neem, both azadirachtin based and non-azadirachtin based. So let's say the compound which is having pesticidal properties as azadirachtin. Method is, and we are extracting by the known method. So method is old, material is old, and if it is used for pesticide, then even application is old. And if you can make it yourself, or you can extract it, you can make it liquid, you can sell it in the market, or you can make it yourself. So this is not novel. But it's the same as azadirachtin, and one of the patents is on that. Long time ago, we discovered for growing hairs on the baldy hat. Now, that's a new application. Correct? So that is novel. When aspirin was used for headache, you have heard about that, isn't it? But aspirin is used for something else also. What is that? Anybody knows? What else aspirin is used for? Please unmute and tell me. One of you should know.
Sir, blood thinner.
Correct. So old compound made by old method, but when it was found first time, it was a new application, which is blood thinner. That was novelty. Correct? So application can be new or method can be new. Instead of using, let's say, normal extraction method, you may be using nano quantity of it. That's the new method. Material may be same, material may be new. Method may be old, material may be new. Application may be old. So instead of azadirachtin or a neem, you use custard apple seed. And let us assume for a minute that it is not known in the literature. When the first time it would have been discovered, that material, new material would have had same property, which is pest control. Same method, let us say, by boiling and extracting, but material was new. So one of the four should be new to be called as innovation. Correct? Is that clear to everybody? Is it clear? Any question about this? So when we document, at that time, we may not know, is it novel or not? But the role of BIIS, what is the role of BIIS? Biotechnological Innovation Ignition Program. Correct? So when you make your projects, when you do your assignments, please do prior search. Please try to figure out what may be the novelty. Novelty can be, again, in several aspects of it. So let us remove it. Okay. It can be in a mechanical solution and mechanical device. It can be in the form, or it can be even compound can be in different forms. It can be in the form of tablet. It can be in the form of vaporizer. It can be in the form of liquid, form, feature, and function. Correct? So novelty can be either in the method, material, use, and application, and the business model. Novelty can also be sometime in the form. Some of you are pharmacy student, I assume, and you would know that the size of the particle makes a difference to the way if we give it by injection, let us say, how much time it takes to disperse in the body. So, for example, one of the famous compound for pain relieving, what is that compound? Do you know that? Very famous. Almost all creams have that. Anybody knows? So, anybody knows what is that compound? Anybody? What was the compound? Very popular. Almost every ointment will have that. Dico? What is the name? What is the name? Please tell me. Anybody?
Diclofenac.
Right? Let me switch it off. So diclofenac compound, injection of this compound, when it is given to the people earlier, it used to cause pain. Some of you might know, some of you might not know. It used to cause pain because dispersal. Anything urgent? Wait कर सकते हो? So इसमें से जो मुड़े हुए निकाल देना. So the that was a problem. An injection for relieving pain causes pain. So what did Ketan Patel do? The main director of the Troika Pharmaceutical. He made the particle size so small that it dispersed in the body very fast and did not cause pain anymore. They patented this in more than 60-70 countries. And Anapar is the compound, is the brand name under which they sell this. Same compound, old material. Synthesis is by old method. Sorry, application is for the old purpose, relieving pain or reducing inflammation, but method was different. And that made it novel. Business model was also old. So why I'm saying this is that we need to figure out where the novelty is. It's in the form. Now they have made a B12 injection. Normally B12 injection also causes a lot of pain. So they have made a nasal spray for B12. Now that's a new feature. A new function is old. Form is old, liquid, but ingested through nose. Delivery system is new. Correct? Otherwise, you have to take it by injection or by tablet. So please understand that the novelty can be in various ways. So when we document by talking to people, we document through competition, we document through Shodhyatra, we document by talking to service providers, we document by talking to the communities. But in every case, we will respect the intellectual property right of the people. If the knowledge is already in public domain, then IPR do not apply. Then you cannot put restrictions. So in prior informed consent, we will share. Amar, please share with them both the forms that we use. Anamika would send you. Anamika, can you send them to the lab, the PIC forms that we have developed? PIC form, so we will send you the form that we have modified for UNDP for their global purpose. And you can again see whether there is a scope for improvement. Prior informed consent. So when we document, we also take their consent that in case it is unique, then rights will belong to you. If it is already in public domain, then the rights will not apply. Okay. So what else we should take care in the documentation? If there is a sample possible, there's a plant which is not endangered and endemic. If it is endemic and endangered, we will not collect a sample. Please understand that. Never collect a sample of a plant which is endemic and endangered. Endemic means found only there. Endangered means there's only few samples, only little population of that plant is left now. We can't take away. So we will have to find non-extractive ways or non-destructive ways of research. We can take leaves, maybe, we can take maybe flowers, few flowers, but we will not uproot. Supposing roots are required, we will not. Or maybe there's a plant which has dried out, then we can use the root of that. But a live plant, we will not uproot because it is endangered. Correct? So we will follow the eco-ethics, ecological ethics. We will also follow the social ethics, and we will follow the institutional ethics. We will be the most responsible scholar. Many times what will happen when you document, 3 or 4 people claim that we know this. All right. You will document the name of all the 4 because you don't know who the first is. Then you ask, once we were in Shodhyatra in Karnataka, in Kopa region. So one fellow, we were talking to one elderly person who was telling us about solutions that they use for herpes, which is frequently find that infection is found in that region. So one young fellow got up and said, why are you giving so much importance to this old man? I also know this plant. We said, all right, just wait for a minute. Then we asked the people, when you need this medicine, who do you go to? You go to this young fellow or you go to the old man? People said, we go to the elderly person because he knows how, not only what the solution is, but he also knows how much to give, in what dose, in what form, what prevention, what precaution we should take, etc., etc. So many times it is not just the knowledge, but also the ability to practice the knowledge, which has to be also documented. You understand? Many people know, but they may not know how to practice it. Correct? There are bone setters, and they may use, let us say, hot jor, very famous plant. It grows near trustee office also, a creeper, and well known that it has a bone healing property. But in what way will we use it? How will we make the paste or boil it or make cold water extract or dry it and make use powder and then boil it? There are different ways of extraction, isn't it? You dry and then extract. Cold water, hot water, soaking, non-soaking, as I mentioned in the beginning. There are different ways. We will, my friend Vivekanand will explain to you different. There's a decision tree. I will share with him. There's a decision tree, choices that are available to us. Multiple choices are there for extraction. And we will discuss in one of the sessions as to how a Nobel Prize was given to a Chinese scholar for Artemisium by using 700-year-old knowledge about method of extraction. They were not finding success. I'll tell you that story later. So we will document, we will do novelty search, we will do the prior informed consent, we will take the prior informed consent, we will try to find who knows and who can practice it, correct? And we will also then see whether this is new knowledge or improvement in the existing knowledge, or it is a traditional knowledge, but not in the public domain, or traditional knowledge which is in the public domain. What is the meaning of public domain? That means it is available on the net or in publicly accessible books or databases. Then it is public domain. Public domain means anything which is known to most people, at least accessible to most people. They may not know it, but it is accessible to most people. That's called public domain. Clear? Any question that you have? We have 5 minutes, we'll ask. Any questions that you have? I've discussed broad issues, which all of you can try to practice. But any doubt, any clarification you need? Dhruv, Murli, Mohan, anybody? Ria has joined back. रिया तुम गायब हो गई थी शुरू में, क्या हुआ था? रिया क्या हुआ था? क्या तुम बीच में चली गई थी ना?
नो सर, सर मेरा डिस्कनेक्ट हो गया था, आई थिंक सम नेटवर्क।
नहीं, हमने आपको रिमूव किया था क्योंकि आप जवाब नहीं दे रही थी रिया। आप डिस्कनेक्ट नहीं हुई, हमने डिस्कनेक्ट किया था आपको।
अच्छा, नो सर, एक्चुअली आई जस्ट गॉन टू फिल माय वाटर बॉटल, तो उसके बीच में हो गया होगा।
अब मैं उसका तो जवाब नहीं दे सकता हूँ। अब हो सकता है, यू मे बी राइट एक्चुअली, बट वी कॉल्ड यू फॉर क्वाइट सम टाइम। 2-4 मिनट तो हमने बोला ही होगा, तो नहीं आया तो फिर हमने हटाया। लेकिन मैं आप सबसे रिक्वेस्ट करूंगा कि व्हेन इफ यू हैव अ प्रॉब्लम, put on a chat that I have to go out, I'm not be able to next 10 minutes, I have something urgent to do. Fair enough, you can take leave, but inform Amar को बता दो, this coordinator को बता दो कि you don't have to announce to everybody, but just inform the coordinator that I will join. I'm leaving for 10 minutes, I have urgently some emergency or whatever, and I will come back. कोई बात नहीं, we can understand that, but
yes, I put it in the chat box.
Connection on and not being there is not a good thing, ना? ऐसा नहीं करना बच्चों. Because
Yes,
sometimes I may ask a question and if you're not replying, it doesn't look nice. Particularly guest faculty होंगे तो और भी बुरा लगता है ना. तो please try to take care. कोई बात नहीं, आज जो हुआ कोई बात नहीं. But in future, please take care. Just inform.
Yes, sir. Sorry, sir.
कोई दिक्कत नहीं, कोई दिक्कत नहीं. Anybody has any question? You understand why we are documenting this knowledge? Why we are scouting? Is it becoming clear now? Because unless you know the purpose, you would not have the excitement and enthusiasm, you know. You should know why we are doing it. It's a very important thing that you're doing. And this is not something that should stop with this. Even later when you, in your professional life, in your personal life, you go somewhere, Sanchi mentioned she went to Chittor and there she discovered, you may go to some function, social function, and you may discover, you can write to us. You can write to lab@sachi.org or BIIS@sachi.org. Tell them that, look, we discovered this knowledge. We want to share with you. It may be interesting to other people. Next batch of this. We will share with the next batch with credit to you. Correct? So please keep this at the back of the mind. Lifelong learner. You are not just learner of the BIIS program. We are trying to induce you to become a lifelong learner. Our quest should never die. It should never stop. It is not a course which is going to end. Physically, it will end on 6th March. And we have got commitment yesterday from Dr. Shekhar Mande, Secretary DSIR and Director General CSIR. He will deliver the validatory address. And we are going to share with him your assignments and your project report that you will write. So mind you, you are going to get feedback from one of the most accomplished scholar of our country. Right? That's what our job is. And supposing he says that, look, this is something, this particular project or that project is something very novel. I would like to offer that person space in my own lab. He has a National Center for Cell Science in Pune, where he was working before he joined CSIR as Director General. That will be a privilege. And we would like that opportunity to open for many of you, that right after undergraduation, graduation, you get an opportunity to work before you join your master's or you become an entrepreneur in the top scholars lab because of your work, because of your insight, because of the rigor with which you explain your problem and try to solve it. So I'll stop here. Any question, 1 minute, and then I will hand over to our next speaker. Has she joined?
Yes, Professor. She has.
Okay. So our next speaker is an entrepreneur. She will share with you her own experiences. How did she was also a scholar like you. Then she came up with an idea, developed an enterprise. So she will share her story with you, and you can, of course, ask her many questions. All right. So I will not take more time and hand you over to Amar. Thank you. And you can always write back any question, even if now you don't have, but if you get in the evening, you can send a mail and my colleagues will pass it on to me and I can come back tomorrow and then take a time in between some sessions or after the session at 1:30. We can even have a separate session in the evening. Don't worry. I mean, if you want something to be discussed in more detail, make demands on us and our colleagues will facilitate it. If supposing 10 of you want a special session on some problem, tell us. We will try to arrange it as far as possible. Okay? So please take initiative. This course is, don't give us feedback only at the end of the course. If you could have done this, that would have been useful. Now tell us now, then we can make it useful when you are still a participant of the course and you can shape the course. We can try to bring in new speakers experts on the topic on which you want to learn something more eagerly. We may not be able to do it for every request, but wherever we find 8, 10 people are interested, we'll do something. We'll try our best. All right. Good luck. Thank you.
Thank you.
And keep trying your assignments well. I'm hoping that by Sunday you will send us these assignments. Kab tak time diya? Sunday ka diya humne?
Sunday. Yes.
So please, and when you submit whatever knowledge that you do, please do some research now. Okay, you understood? Do some research, find out. You can have 10 references, 20 references, we don't care, but find out why there are 20 people working on the same thing. Are they repeating the same thing, or they're finding different aspects of it, different functions of it, different features of it? Then you will learn how to do literature review. You will learn how to read papers. Because tomorrow when you have to write paper, you should know how to read paper. Before writing, you should know reading, and it's very important art. We'll discuss it later. Good luck. Bye-bye.
Thank you, please.
Yeah.
Dr. Vanitha?
Oh, yeah. Can you hear me?
Yes, doctor.
Yeah.
Yeah, the floor is yours, doctor. You can talk with the students, interact with the students.
Oh, good morning. Good afternoon, students. Sorry for I am being traveling and the internet connection was not stable. So sharing presentation will be difficult. As sir has mentioned, I am CTO of Ravi Environment Solution. And my journey starts with a biotech student who was doing her PhD with allergens. And when I went to Patel Chest and I met the patients for collecting the blood sample, I came to know about the condition they are living. So their sanitation condition poked me that what they will do with the allergy. They don't have the basic immunity, and kids are dying just because of diarrhea. So I should do something in line to do those conditions. These common man needs to have some standards, and there should be some options. So I started working on waste management rather than allergy. Immunologically, I was developing an allergy kit. So I changed my PhD topic after 6 months, and I started researching on what is the waste, like what is the sector all about, and how I, as a biotechnologist, I can help. So I converted myself to an environment biotechnologist, and I done my PhD on that. During that time, I could able to convert this waste into hydrogen, which is a future fuel. And nobody was talking about that thing at that particular time. It was way back in 90s, late 90s, 98, 97, I was working on this. So when I got the patents, nobody was ready to accept hydrogen as a fuel and all that stuff. So I worked as an R&D head of an environment consultancy, where I could solve the short-term problems, that is ETP, the wastewater which is going and contaminating. I started working on that as an R&D head. So we done a lot of ETPs and all. And then I came to know that there are other products which is required, that is these microorganisms which are catalysts for the biological systems. So I could able to provide these companies with these cultures, and my company used to use a tagline. We provide tailor-made cultures. So I used to do whatever I could do in the environment consultancy lab, which is not a biotech lab at all. Whatever I could able to do, I used to provide those kinds of cultures to them. Then later on, my director also told me that whatever you provide, it sustains the ETP. So why don't you protect this as a product? So I tried to do it with the company, but I could not able to, because there was a lot of funding required, which was not there from the private institute or the private company. So later life moves on, and then we got these incubators. There is one incubator in Baroda where I am incubated right now, has come up. So when I came to know about this, I wrote this particular project that I wanted to have these kinds of product under BIG grant. And I am fortunate enough that this project was approved for funding in first go. And I got that funds, and I had a space in incubation. in this new incubator. So I resigned from my job and started as a woman scientist with this particular idea to validate. So after the validation, I started the company. So in short, the journey is from being a scientist to entering an industry and then coming back to make your own company. So it's a long journey. So I'm very happy to answer your questions, whatever you have. This is the short background I have, but during this whole journey, I have come across a lot of schemes, the ecosystem which has been developed around us to thrive the innovation, which was not there in 90s or the early 20s. So that is the change in the ecosystem, and you are lucky enough to have this ecosystem and take most benefit of this. So if you have all the questions, I think, sir, if this session can be more interactive, I will be more than happy to answer the questions.
Students, you can ask whatever queries or clarification or whatever you are having doubts.
If you want to know, understand some schemes, I can answer those. If you want to understand how an environment biotechnology can move around, I can answer those also. I was listening in the last, sir was talking about some plants and all. So anybody of means among you is working on to environment field, they can ask the questions.
Anyone who is interested in environmental biotechnology?
Not only environment biotechnology, if you want to ask anything on the biotechnology, it's more than okay to answer.
Good morning, ma'am.
Yes.
So I had a question that do you feel that some sort of experience working as a researcher or as even an intern, like not intern is definitely a good thing, and like as a researcher, like after you finish your education and all of it, is it required that you first do a research, like be in the field, experience the field, and then it is better to start your own company with your own vision, or is it okay if you start from day one?
Answer to this, generally in my slides, I show that there is no age bar or no limitation. If your idea is different or you really want to explore it, you get mentors around you. You can start it at any point of time. You are even in the school, you can start. There is no such age. If your idea is having a weightage or it's really solving a problem, you will able to demonstrate it. Then you will get a lot of there is a lot of ecosystem help with which you can grow. You can have your team members across you doing your SWOT analysis that this person I need. So there is no need, no such thing that you have to do this thing and then only you can do the innovation. No, you have to be very vigilant that you are thinking or you can identify that this is the problem. When you are doing a research, there should be a problem statement around which you are working, correct? Sir was mentioning that there are 20 people approaching that particular thing, but their way of approaching may be different. They will not be doing the same thing when they are doing the research. So identifying your problem and how you will be going to solve your problem, that is where you have to have, if you got that idea, if you got that, but that question should be in your mind. Like I was, when I started on waste management, I never thought of, till my MSc, I was very passionate to work on immunology. And I started working on allergy kit. I was developing allergy kit. I was doing IgG, I was doing IgM. And I was isolating from the blood. But when I got this blood sample and I went to meet those patients with whom blood samples I have taken, and I found them allergic
to these allergens, then I means the things have changed for me, that ki jo main kar rahi hu and what these people are living conditions are so bad. What should they will do with the allergy? So those are the things, once you get something which is very close to your heart, you might get your answers. Have I answered your question?
Yes, ma'am. Thank you so much.
But acquiring skill is equally important. You believe me? Still, I am on as a CTO. I acquire skills like business front. I was not knowing. You know me as a researcher till I started this project as a woman scientist. I was not having any business skill. So for last 3 years, I am trying to acquire those skills like terms in business, how I will do. As a founder, I have to think very differently. So acquiring skill will never stop, You have to acquire skill. How and from where, that is another issue. So acquiring skill is ongoing process.
Yes, ma'am, that is what I was asking, that, because like when we are young, we probably do not know as much about the industry as it works. So if we grow a little older with a little experience, we might be able to do a better job at creating our vision.
That's what I'm trying to tell you, Ayushi. What happens, like when you are very young, you started working on some of the problems. You are solving it scientifically. You are a researcher, like biotech researcher. I was a biotech researcher. I was not knowing the business, but there should be some solution which is justified by some other people कि ये जो solve कर रही है ये ठीक है. This path is correct. Then you will get mentors to mentor you onto the other aspects of that particular thing to take it to market. Correct?
Yes, ma'am.
So you have ecosystem where you will get mentors, like sir was mentioning that you need this thing. You have to identify that, okay, I am going to this path and I need this thing also. Like suppose we board a bus and I wanted to reach some place, but the journey is for whole day. I need water, I need food to sustain. So I have to figure out, either I will have to carry it myself or I have to get it somewhere. Correct? But you have to identify that you need this thing.
Yes, ma'am. Now I understand your point. Thank you so much.
Yeah, you can write a mails to us that we are solving this particular thing. This is the solution and we need these expertise. So right now I am mentoring a lot of students who has formed a company. They are researchers like you. So I can guide you onto the technical side. And as an entrepreneur, when I am doing converting from a researcher to this, I have done a lot of things which I have learned. So I can give you those skills also in a very short. We will not be experts on that. We will be needing an expert like CS. I don't know. Company secretary, I can't be. But now I understand what I have to get from that company sector. Correct? So those kinds of things also you can ask to us.
Thank you, ma'am.
Right now, like one of the person who is doing a biotechnology research, they want to scale it up to some level when they are doing any process. So in that case, we can provide you a technology mentoring that, okay, you have demonstrated into a Petri plate or in a flask that this is happening. Or like if you are extracting some herb, we can help you to scale it up. Any more questions?
Any questions, students?
As a girls also, you can ask me how difficult it is with the social structure to move around.
think the students at least respond something. No?
Hello, sir. I'm saying that this is now stable. So show the slides also. If they want to know more about Ravi, I can just scroll the presentation also.
Yes, doctor, you can. Maybe if they see some slides, they may get some
Yeah.
idea.
So can you see the slides now?
Yes, doctor, it's visible.
So as I mentioned, the story is from 70s to 17, where I have founded the company. I'm born and brought up in a Punjabi family where girls' education was not important. My dad, I have to get scholarships all the time to continue my education. This is the thing which has changed my life, because I got to reach this particular conference where I came to know about BIIS grant, and that has changed. Give me a platform for entrepreneurship. I resigned. I never thought of that I will be an entrepreneur. I have never thought of that I will be an environment biotechnologist first. Then I have never thought of being but there was a problem which was hampering my mind, that how I can help as a biotechnologist to improve this particular condition of these people. So that's what I usually say. You have to believe your intuition. If you have something which is triggered inside you, you will find a way to do that. So this is the ecosystem right now we have. You name it, what you need as a resource can be provided from this ecosystem. So as biotech startups, we have been given a lot of opportunities. I am a DIPP recognized startup where we get all these benefits. So there are a lot of benefits which is all around. If you have really solving an major problem, you reach out to us, you frame it properly, like sir was mentioning, document it properly, and then we as mentors can help you to present it in various schemes and take it. Like I think all are in early stage. So there are these schemes which are available, not only in this, but in Gujarat, you have VT policy. There are other things you will be getting, especially for the women entrepreneurs. There is a special platform where you will get a mentors like us who have done something in the entrepreneurship and can be your partner in this lonely path. Sometimes we don't get a woman to talk to because there are other issues happening around. For me, people have told me when I started this entrepreneur journey that you are making a professional suicide at the age of 40. So that was the thing. But I thought it is never too late what you might have been. So these are the younger one, you know, that's what I am mentioning. They are the CEO of the company. So you can think of कि अगर ये CEO बन सकते हैं, so there is no age limit. And this is the person who have started so late. And we all know these two people. So like I was mentioning why I started this environment biotechnology when I started understanding waste. During that time, we are just coming across the ozone hole. So I understood that there are a man-made disasters which we have already done. On the terms of development, we have all this.
Doctor, your slides are not moving, I think.
Not visible?
No, it is visible, but the slides are not moving. It is in the first slide itself.
Oh, sorry. I have moved on to so many slides. So now you can see?
Yeah, now. Yes, yes. Doctor.
So should I keep it in this mode only?
If you possible can go to full screen mode.
I have been in full screen now. Can you see? I changed the slide. Can you see?
Now in the second slide it is.
No, when I move to this full screen, it is not moving then.
Okay, then normal.
I can go ahead with this slides.
Yes.
What I was mentioning. This was the letter where these are all schemes which I have listed. If you can see this, if
now it is working.
Yeah, these are all government schemes which I have listed. These platforms can be reached for knowing more details about this project, these schemes. So this is especially for the women, the girls who are founding their companies, and they will get mentors, specific mentors here if they really want it. So this was, I was mentioning that these are the two boys who have started their company when they are so young. They were in the school. So there is no such thing that we have to start working on our idea at any age. It can be done at any time, but we have to have a clarity that what we want to do and what we need. That is the only thing we need to have. That clarity, that what we are working on, why we are working on, and what we need for that. So you will get it from the ecosystem. So like I mentioned, my hinge started this condition. So I started working on to the waste management, and I came to know that as human beings, we have created a man-made disaster. This was only, we have created it. And now people are saying when we are attending conferences that if humans are not there on this planet will flourish. Correct? So this is the situation. But as human beings, if we have created something which is wrong, as human beings, we can correct it. And that's where I started putting a drop. I will not say that I have changed all the things, but I started putting a drop in this whole ocean to change this particular scenario. So like what has happened in environment sustainability, everybody knows there is a climate change happening. We have to limit our greenhouse emissions. So that's why we are working on renewable resources. So what I could able to figure out is that there is an organic waste which is generated by our population. And people are talking that our population is an issue. But our population is a resource also. So that's why I started working on this particular aspect. And to limit this greenhouse emission, I started utilizing this organic waste as an energy resource. And that's why I worked on to the hydrogen production, which as a researcher, I could find out that this is the future fuel, which creates no greenhouse emissions. So I started producing this particular thing using waste. And now when our Prime Minister announced hydrogen mission, I am very, very hopeful that within I will say another 2 to 3 years, you will be seeing this biological hydrogen. I have worked it on long back, 20 years back, but no ways I had that knowledge, that skill, which can be used by the society now. That's what we are doing. So in my opinion, I say there is nothing a waste. It's only a resource placed at a wrong place at wrong time. We just need to find a way to use it. And that is best out of waste. And we all are doing it from our school level itself. So only thing is we can utilize this particular resource in many ways. Like I am doing biofuel, gaseous fuel, somebody is doing it, ethanol, some is doing requesting, some is doing composting. There are so many ways to approach to use this resource. I will say this resource. So you may come out with some other idea. There are people now using this particular waste for making packaging materials. There are n number of ways by which we can use this resource. So there your creativity, your thinking comes that you want to use this particular resource in this way, rather than having a burden. You can convert this as a resource. That's what we are trying to do. And as society, we have to, we were following linear economy, and that's why we have created all these issues as you understand as biotechnologist or a science student, that there is this nature works on a circular way. We have all the cycles. We have carbon cycle, we have water cycle, all there is a cycle, and the cyclic form is very important. So what we need is to change our attitude from the linear economy to the circular economy, and everybody, scientists to politicians, are working on this right now. So I am also trying to contribute whatever I can. So if you see only the manure available in India, we can convert this greenhouse gas emissions to a resource, and that is where I am working on. And I appreciate if anybody has any idea on this particular aspect, greenhouse gas emissions, developing a technology where there will be a less carbon dioxide emission. It may be in inventing a particular kind of heater which will be using less electricity. There can be anything. It does not mean that we have to stick to this thing, but we might have another ideas. So these are some of the resources. Since I am an anaerobic digestion expert, so these particular materials, if we use, can be converted into organic manure. Along with converting into gaseous fuel, we will be getting this. So I will be contributing into the water food nexus. I want to give back to the society the treated water. At the same time, I wanted to give them organic farming and a energy source in the rural area. That is what I am trying to target. And in India, when we are seeing the the GDP, India is bending upon to the bioeconomy, and the biofuels will be the 6% of it. But there are other things where we can contribute as biotechnologists. You can be an agri startup, you can be a diagnostic person, which I have also started. You can do a vaccine, you can do, but there with the COVID, you know how we can, how much we need as medical support. We see that people need that oxygen supply when the things have happened. So if we have done some basic research, we might can solve that problem, which will come at particular time. So that is where we have to be very vigilant as science student. What are the issues faced by the society? So what I am working on is to the anaerobic digestion, which is the core or the heart of the circular economy. You can do n number of things taking this into in the center. Correct? I want to have know the students' inputs. If I put it here, what they know about this, to make this little bit more interactive. Can I have some ideas? Can anybody? Should I call names? Hello?
Students?
Can all listening? I don't know. Because they might have ideas here. Because with anaerobic digestion, you can do bioplastics, you can do ethanol, you can do a lot of things. From here, I want to know from everybody, what are their intentions of work right now with this course which they are doing. So I will start from the participant list.
There is a question in chat box.
Okay. Wow. Great. Fermentation product, ma'am. That is the answer. How to develop anaerobic culture to utilize agriculture residues. Okay. Great question. I am working on this for the anaerobic digestion. What we need is, especially for agriculture waste, it can be easily biodegradable. So, a normal methanogenic culture which we have can convert anything in any agriculture residue. But to get maximum out of it, we have to open up the lignin structure. So there are two approaches. Either we have to take out that lignin separately, because lignin is a polymer which can be used
So I am working on to the two different approaches, wherein one is the same thing, that I believe that lignin is a very strong polymer. Rather than degrading it, if we can utilize it in some other way. So what we have done is that we have removed these lignin fibers from the other portion, and one portion we have taken for the biogas, the rest we are trying to convert it into cellulose or the nanocellulose. That is what we are trying. But farmers have this issue because they are burning this waste. We all know that they are burning, and Delhi is facing a lot of issues. So right now I am working on another project where I am trying to make the compost, wherein I am using other, not bacterial cultures, not methanogenic cultures, to decompose this particular thing. Something has been done by PUSA also. They have done this microbial cultures, but in spite of that, it takes around 20 to 25 days, and farmers doesn't have this time period window. So they want something which can be done quickly, maybe in 15 minutes, but I could not able to reach that point. That is a challenge I have, but I am trying to do that particular decomposition in 10 days. This is the challenge I am trying to solve for last 6 months. And we have done trials last year, and farmers were quite happy getting those. So mixing cultures, isolating anaerobic cultures, there is a normal pathway that you have an anaerobic condition where you isolate those cultures. Normal isolation way of anaerobic cultures. There are now techniques to do the bulk thing. And when we isolate a consortia altogether, we have to take help of metagenomics to identify those, rather than doing a single microbe kind of thing. Have I answered your question?
Yes, ma'am. Thank you, ma'am.
Yeah. So what fermentation products can be made by this particular thing? Because somebody has answered this. To elaborate on these fermentation products, which we can do with anaerobic digestion. If you take anything like I am mentioning that we can do bioplastics, we can stop at one particular step, and we have this particular thing, bioplastics, the monomer for that, and then we can make the bioplastic out of it. You can name some more products. The answer is correct, that fermentation thing, but you need to elaborate. You have to see what other things can be done. There are some things mentioned over here, but you can do a lot of things. And when we be having this particular waste, like we have extracted oil and then there is left out material, there can be skin, the cosmetic things can be prepared from that particular oily thing. There can be health benefit things for animals which can be prepared. It can be for humans also, but for the animals also, because they are not have, there is a lot of things needs to be done on animal feed. Correct? There can be a fruit juice extracted from some of the fruits, and then this pulp is there. Correct? Hello? Yeah, there is some message. Agri waste in production of bulk amount of industrial products, ma'am. To give examples for this agriculture waste, I was mentioning there are people who are using this agriculture waste. There is one startup, if you know, Fool, who are using the flower petals to make some other thing which is looking like leather. That is a fermentation technology only. There are one other startup who are working on to converting rice husk into, they are doing a lot of things. They are doing some biotechnological intervention, and then that is converted into a packaging material which is biodegradable. So they are placing thermocol with that particular product using some aspect of biotechnology. That is where they converted into some product, and then they have created a mold with which they are converting into different kind of packaging materials. People are using it as a this thing, disposable cutlery jo ban raha hai, they are using it
The agriculture waste has been used in that particular thing also. Yes, you can produce bioenzymes, but which kind? You have to use microbes, but this waste material might, these are cells. You might get something, some particular enzymes, or growing some other microbes on this particular waste, you can get a lot of enzyme. People are producing xylanase, people are producing lignin peroxidase. Lignin peroxidase is one of the enzyme which we need. We need in a waste treatment or somewhere, but the cost of producing lignin peroxidase is so high that we cannot use it. Cellulase has been produced, pectinase has been produced, lot many enzymes have been produced. So you can do that also, correct? You can produce any bioenzyme, you can produce catalase, you can produce any other enzyme. If you can optimize those conditions with a cost effective manner, then that can be that can reach to market. But if you can produce it in a very small amount with very high cost, there will not be any customer paying you. So that cannot be a business solution. Anything else? So, okay, I will
Other
go.
questions, students? Yeah, one more is.
One more thing came up. Can we extract biofuels from cooked food? Yes. Yes, we are doing. We are doing kitchen waste. There are lot many decentralized plants, and we are converting that cooked wastage which has been there from the kitchens or the mess to fuel. We can convert it into hydrogen. We can convert it into methane. So for automobiles, yes, CBG is produced using this food waste. For buildings, what options you want? Like we are providing it as a fuel, or we are converting it as a electricity, whatever biogas has been produced, and as well as CBG. Right now, I am trying to work on biohydrate. That is basically where we will be mixing hydrogen and CBG, and we are trying to figure out how that can be utilized where engineering aspect is required, because there compression, their storage and all that stuff apart from production. As a product, when it has to reach, we have to do all these. Ma'am, I have done bioenzyme fermentation product by using jaggery and citrus waste as substrate. Great. Which enzyme you could able to isolate? How can know about the microorganism responsible for that? So if you are you want a cellulase or cellulase enzyme, then you have to search for, when you will be searching, you can search for cellulase, microorganisms who will be secreting cellulase. You will find a list of microorganisms. You can get it from MTCC. You can isolate it from any of the source, and then you have to do the characterization of those cultures. And when you will start doing these cultures isolation, you might reach to the same microorganism which is already submitted by somebody else in MTCC. So you can just see what exactly you are doing and from where you are getting these kind of source. You can unmute and ask if you have some specific thing.
Yeah, for cellulose screening, they can use CMC medium, cellulose degradation.
Yes, you can use.
It will be easy to identify the organisms. Yeah.
Yes. But you have to see till which point people have already done it.
Yes.
There you have to take it. Otherwise, you will be just spinning the wheel again.
Either you want to use the whole organism or you want to isolate the enzymes and use the enzymes for applications.
Okay, which question I was in this fermentation? Yeah, I answered this. Can we produce bioenzyme using yes extract Ethanol is produced by fermentation directly in the petrol to make biofuel. Directly, it is not directly produced. Bioethanol is produced using agricultural waste and then it is blended with the petrol.
Ma'am, we lost your voice.
Lost my voice. Hello, can you hear me now?
Yes.
Maybe again the internet issue might be coming. So I will go through these slides and then we can discuss. So like similarly, when I have talked about the microbiology, there is an aspect which is hardware. There has to be design of the reactor. When we are doing a fermentation, the main thing is in which type of fermenter we are using it or which type of reactor setup. So there are a lot of research can be done on this particular part. Which type? So that is also wherein we need a team, like an engineer in our team. If we have a combination of this, we might be getting a better yields. So what we are doing at Revi is we are trying to do these biocultures, and we have done this particular India's first product, which is methanogenic culture. And that has been validated using a lot of different waste, and that has been validated by third party that, okay, when we use this, we are reducing our COD or BOD, that is, we are reducing the pollution reduction. Because I am working with this wastewater treatment also, there is a lot many effluent generated, and I am working with them to treat their effluent and use that water back. So that is where these parameters are very important. So if we are producing acetic acid, that can be a product which I can utilize rather than converting it to methane. There are many other things which we can do, but I have mentioned here propanic and acetic acid reduction. But for methane, I have to do that. But this can be a product if I can stop it in between. And we are working on that also. So these are the performance when we have worked on to the product, not only in the lab, we have to go to the customer side and see that if this product can work there, because these are microbes and there is no sterile conditions there, how they will be reacting there, that needs to be established. So we have revived anaerobic digester, which was not working with our cultures within the first week, and they were very happy, and they have given us this particular what do you say, the validation of our product, that this can be a product which a client will buy and that will solve his pain point to treat the waste, dispose the waste. And that's how this product have been validated, and now we are selling these as this particular packaging, adding to the all the SDGs goal. These are the SDGs goal our product is taking care of. We are doing circular economy. We are giving enhanced bioenergy. Most important for the client is we are saving their treatment costs, although that is not an SDG goal, but because they are saving their operating cost, that's why they are buying this particular product. And in when they buy this, they are reducing their greenhouse emission. And that's why the industry gets benefit out of it. They are getting reusable water. And that's why they are very happy with happy using this particular product. So that's where the customer perspective comes in. So our technology, this is the technology comparison slide. When we see that wastewater has been treated by aerobic thing, what is the advantage using anaerobic, conventional anaerobic, but when they use Revi anaerobic culture along with their hardware, then these are the benefits. And that's what are happening. So when we have compared our product with the existing products in India, these are the product, even the cow dung, because you know, if to start an anaerobic reactor or gobar gas plant starts with the cow dung, and cow dung has a known fact they have methanogens. But you can see in our product we have almost the double archaea in our product. So that's where the developed consortia by us is more valuable. So this was our journey. I mentioned to you that I started in 2016 and we are here. So it took us a lot of time. This is a pilot plant which has been funded by BIIS to demonstrate that we can produce, we can scale up the production of this particular consortia, and still we can. And after that, we have supplied it to these customers, and I have shown the validation done by them that cultures are working. So we got orders worth 2 crores from people. They want to buy this, but the happy problem we are facing is we don't have a reactor where we can produce this much amount of cultures. So we are looking forward to get these fermenters erected. Maybe within next year, we could able to solve this thing. There are some awards which have been given to us. This is the motivation for me to work. You can interpret vitamin M as money also, or you can say it as motivation, because these are the real things when I could able to demonstrate not only onto the scientific part, but as the business part. After winning this particular biotech winner award, and this year only we got the National Entrepreneurship Award, we got motivated to work further. But the challenges are a lot. Incubation space. Right now we have to graduate from our incubator, and we have to have our own R&D setup. So a lot of not only money, constructing that building, doing all those stuff is a challenge. Apart from what I am doing on the science front, I have to manage all that. Then because of COVID, whom we have hired, they left it, and then we have to change the whole team. So maintaining team for any startup is a difficult task, not only for us, but the corporate also have to have this trouble. Then every day there is some procurement required, from buying a pipette to buying a reagent for doing the experiments. Then there is equipment breakdown. Aaj centrifuge nahi chal raha, kal autoclave nahi chal raha. All those are the issues which you have to manage when you will doing your startup. There will be a financial crunching. There are orders to us, but we cannot supply because we don't have a financial resource to erect those many things. Then if you have outsourced some work, your report have not come, or that has not been done, then there is always a meeting deadline where we have to, like sir was telling you, to submit the assignment. When we are getting some funding, we get it with the milestones. We have to give them that. Even for the awards which have been listed, there are forms, there are pitches, there are everything. So we have to meet those deadlines. Then apart from that, we have to file the IPR, we have to maintain those IPR, we have to answer the query coming from those. Although the process patents have been granted for us, so that has been done, but that has to be done simultaneously. So those are the challenges. It is not very easy, but only thing is team and coordination. That's what I mentioned. You have to figure out what you need from whom also. You have to figure out, and then you have to reach to them. So this is our team. Me as a CTO, I am biotechnologist. Rajneesh is an MBA, and he's looking after the business development, and Bhavik Ji is an account person who looked after to the GST, invoice, you name it. There is a lot of issues. There are which we have to file every month. Then there is a CA audit. Then there are CA things. So Bhavik sir used to see that. So then we can work. So our aim is that we need water for all and a revenue of 200 crores in 5 years with a green earth. That is what is our objective. And we are really working hard to achieve that. And as I mentioned, this is a roller coaster ride, which I am following. Every day, it's a different challenge. One day we are having a fall. We don't have anything. We feel like that we really cannot survive for the next day. And immediately there something happens and we are again up and then again down. And this is happening for last 5 years, I will say. And that's what I want to share with you all. Thank you.
So I stop sharing here. Yeah. There is one question, I think, huh?
Yes.
Yeah. Ma'am, when it comes to green energy, people and government body only thinks about the high initial cost. Agreed, but there are now, since we are talking about a lot of things on climate change, there is an impact factor. There is amount of greenhouse gas emission. There is a carbon trading coming on. There are many things coming up with the policy change when we are talking about green. There is a cost involved. There is a very high cost involved, capital cost. But that is what I mentioned. If I am reducing the operational cost, as I shown you, when I am comparing it with the aeration technology, they have an operational cost. Every day they are using electricity. So when they are treating that particular thing with an anaerobic technology, they are saving it on that side. And that's why that becomes an ROI for the client. So that we have to understand. We as innovators have to calculate, we have to show them. So there are other impacts. So there are now impact fundings coming up. But we have to show that whatever we are developing has an impact, and where and how much, that has to be calculated. So that justifies your initial cost. Hope I answered. Could you, what is the name of that person?
Tarmoy Das.
Tarmoy Das. Tarmoy Das, do you have anything in mind? You can ask the specific thing by unmuting yourself. Thank you, sir. Then if no more questions, we can end this session.
Yeah, thank you, doctor. Thank you so much for giving us a lot of views about your journey as a woman entrepreneur as well as a scientist.
That's what I am saying. If these kids have any specific question and working on any of the biotechnology aspect, I am more than happy to be connected. They can reach out to me. You can share my email ID with them.
Sure. I also having 2 students working on a lignocellulosic degradation. Maybe I will ask them to write to you. Maybe.
Sure.
For their master's project, they are doing 2 master's project is going on. So I will ask the students to write to you. Maybe they can visit your facility also. They can get an idea. You can help them.
No issues. They can come up. We are working on this compost project.
Yes.
There is one intern who just joined and we are giving them a training kind of thing for 2 months. So people can also take benefit of those kind of.
They are also focusing on.
But there is very limited space we have, so we offer 1 or 2 students only at a time.
Okay.
Yeah.
Thank you, doctor. Thank you very much.
So can I log out now?
Yes, doctor.
Thank you.
Thank you so much. Okay, guys, that's all for the today. Other things I will share with you in our WhatsApp group. Okay, regarding the assignments, everything we will discuss in the WhatsApp group. Okay, thank you for participating.
Thank you, sir.
Thank you, sir.
Yeah, thank you.